को होस्ट करता का? जवळ साहेब, मी कनकरे सर यांना को होस्ट करा.
फक्त सर्वांना विनंती आहे, आपण लाईव्ह जातोय. कृपया आपले व्हिडिओ ऑफ कराल का प्लीज?
Sir, good morning.
Good
sir,
morning. Good morning. Good morning.
सर, सर्वांना विनंती आहे, कृपया आपापले व्हिडिओ ऑफ करावेत आणि म्यूट करावा. आपण लाईव्ह आहोत आणि पुन्हा एकदा हा व्हिडिओ प्ले होईल आणि मग आपण सुरुवात करणार आहोत. धन्यवाद.
नं बहुकुर्वित तदव्रतम्.
जय 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 हरित क्रांती, जय 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 नवल क्रांती, हरित क्रांतीचे नवल क्रांतीचे कार्य जे करीतीनी, जय महात्मा फुले कृषी विद्यापीठ.
पश्चिम महाराष्ट्र खानदेशात पसरविले कृषी उत्पन्न जाळे.
आधुनिक कृषी तंत्रज्ञानाने निर्मिती भाजीपाला फुले फळे.
सातपुडा सह्याद्री पर्वतरांगांवर,
तापी गोदावरी मुळा कृष्णा काठावर,
विश्वजन कल्याणासाठी रचिल्या अगणित विकास वीर, अगणित विकास वीर. जय 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 महात्मा फुले कृषी विद्यापीठ.
माळरानी हिरवेगार गालीचे फुलवीती,
शिक्षण विस्तार संशोधन ग्राम उन्नती.
अन्न पशुधनाने दौलत वाढवीती,
अभियंत्यांच्या अवजारांनी विविध प्रक्रिया घडती. जगी फडके झेंडामानाचा असे हे गौरवपीठ, असे हे गौरवपीठ. जय 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 महात्मा फुले कृषी विद्यापीठ. जय 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 महात्मा फुले कृषी विद्यापीठ.
कंकरे सर, आपणास विनंती आहे, आपण सुरू करू शकतो. माने सर.
को होस्ट करा.
हो.
Shall we start, sir?
Yeah.
It gives me immense pleasure to extend you a very warm welcome on behalf of MPKV ICRTC Agriculture College Pune, who has clubbed all of us for the brainstorming session on conservation and improvement of indigenous cattle. My heartiest welcome to President of Brainstorming Session, Dr. Prashant Kumarji Patil, Honorable Vice Chancellor Mahatma Phule Krishi Vidyapith Rahuri. My head will tilt in reverence when faced with the genius of a great supporter like him. He is a wizard for our ICRTC project. His sublime thoughts, persistence, and austerity purged this project like nothing. He turned out to be real savoir. He is responsible for the astonishing lyrics and a composer for the fine melodious tune of ICRTC. I welcome you, sir. Sir, you believed in us, which help us believe in us. My exceptional courtesy and extraordinary respect for the guest of honor, Dr. Shyam G. Zawar. He is artistic genius. Dr. R.K. Pundir, humble scientist. Dr. Archana Verma, all are impeccably polite and persons having scientific magnanimity. I also acknowledge the presence with the great pleasure of Dr. Sharad G. Gadak, the Director of Research and Director of Extension, Mahatma Phule Krishi Vidyapit, Dr. Pramod G. Rasar, Dean, Faculty of Agriculture of Mahatma Phule Agriculture University, Dr. Sunil G. Masalkar, Associate Dean of Agriculture College, Pune. These all these people act as a fuel for our enthusiasm, and their kind words are like a fortification of positivity. I also welcome to the technical head, Dr. D.K. Kamble sir, and who is the head of Animal Husbandry and Dairy Science. I also welcome Chief Scientist, Dr. Somnath Mane. And it's so efficient, it's so electrifying, it's so fascinating, it's so rewarding, and mind you, a breathtakingly beautiful that all prodigious panelists, and these are all decent people with superb intention. Dr. Anil Bikane, Director of Extension, MAPSU Nagpur, Dr. Nitin Markande, Associate Dean, Veterinary College of Perbani, Dr. Y.G. Fulpagare, ex-Associate Dean of PGI, MPKV Rahuri, Dr. Sanjay G. Madak-Male, Senior Scientist on AICRP on GOAT, Dr. B.R. Ulmek, ex-Dean, Mahatma Phule Krishi Vidyapit, was ex-head also, Animal Husbandry and Dairy Science Division, Dr. N.V. Patil, Head, ICR, Central Arid Zone Research Institute, Jodhpur. He will also join us in a short period. Dr. T.V. Raja, Chief Scientist, Central Institute for Research on Cattle, CIRC Merit. Dr. B.D. Savalia, Head, College of Veterinary Science and Animal Husbandry, Junagadh Agriculture University, Junagadh, Gujarat. Dr. V.K. Patil, who is having immense help all the time, Co-Commissioner, Central Cattle Breeding Farms of Suratgarh, Rajasthan. Dr. Dhananjay Parkay, Co-Commissioner, Animal Husbandry Division, Maharashtra State. Dr. Ajay Pal Aswal, all the time he's also extending helps to us, in charge animal biotechnology, especially for the Red Sindhi, he's helping us. For Uttarakhand Livestock Development Board, Kalsi, Uttarakhand. Dr. S.S. Lathwal, he is always being with us in the farmers of Sahiwal Club of Maharashtra as well. Lathwal, Dr. Lathwal, Principal Scientist, Livestock Production and Management Section, NDRI Karnal. Dr. R.K. Singh, Professor, Rajasthan University of Veterinary and Animal Science, Bikaner, Rajasthan. Dr. B.G. Desai, Head of AHDS, Dr. BSKKV Dapoli. Dr. B.B. Thomre, sir, Associate Dean, College of Agriculture, Ambezogai. Dr. S.E. Jadhav, Senior Scientist from Indian Veterinary Research Institute, Izzat Nagar, Uttar Pradesh. Dr. G.K. Londe, Head Department of AHDS, Parbani. Dr. Manoj Avari from BAIF. Dr. Sheshrao Chavan, Head AHDS, Dr. P.D. Kavi Akola. Dr. Dhananjay Boite from Baramati. Dr. Shantaram Gaikwad from Fulton Doodh. I heartiest welcome you, all of you. Now, let us have the pleasure of listening to the welcome address from our respected Director of Research, Dr. Sharad G. Gadak, sir. Over to you, sir.
Good morning to all of you, friends. Today we gather here for the brainstorming session on conservation and improvement of the indigenous cattle, organized by the Indigenous Cattle Research cum Training Center, Department of Animal Husbandry, MPKV Rahuri, College of Agriculture, Pune. And on this occasion, Honorable Vice Chancellor and Chairman of today's function, brainstorming session, Dr. P.G. Patil sir, then guest of honor, Dr. Shyam Zawar sir, National Mentor, Ministry of Fisheries, Animal Husbandry and Dairying, Government of India, Dr. R.K. Pundir, Principal Scientist, Animal Genetics Resource, ICR, NBAGR, Karnal, and Dr. Archana Vermaji, Principal Scientist and Head, Animal Genetics and Breeding, NDRI, Karnal, and my colleagues, Dr. Pramodji Rasal, Dean Faculty of Agriculture, MPQ Rahuri, Associate Dean, College of Agriculture, Pune, Dr. Sunil Masalkar, and Chief Scientist and Indigenous Cattle Research Training Center, Dr. Somnath Jimane, and Head Department of the Animal Science, Dr. D.K. Kamble, and all reporters of this today's program, Dr. Mandak Male, Dr. Sakre, and all organized team, and all the panelists. I first welcome to the Honorable Vice Chancellor, Dr. P.G. Patil sir, who has given the very valuable time for this inaugural of this brainstorming session. I welcome on behalf of MPQ Rahuri sir. Then I welcome to the Dr. Shyam Zawar, Chief Guest of Honor, and National Mentor, Ministry of Fisheries, Animal Husbandry and Dairy, Government of India. Sir, I welcome from MPQ Rahuri for accepting our guest of honor today for the brainstorming session. I welcome to the Dr. R.K. Pundir, Principal Scientist, Animal Genetic Resources, NBAGR, Karnal. Sir, I welcome warm welcome on behalf of MPQ Rahuri for accepting our invitation and your presence in this brainstorming session. I again welcome to the Dr. Archana Vermaji, Principal Scientist and Head Animal Genetics and Breeding, on behalf of MPQ Rahuri for this as accepted as a guest of honor, and today is present in this offline mode on this in this brainstorming session. I welcome to the Dr. Pramodji Rasal, Dr. Sunil Masalkar, and all our colleagues, and also I welcome to the all panelists who are present in this offline mode for this brainstorming session, and I welcome to all my colleagues and scientists of MPQ Rahuri for their organization. Then, sir, this is a very important topic we selected for the conservation of improvement of the indigenous cattle and for brainstorming. And this is, the India, our world milk production of our world milk production is about 800 more than 850 million tons. Out of these, India ranks, our country ranks first in milk production, which we achieve 198 million tons of the milk production. Per capita availability of milk is 407 grams per day per capita, and this is a very great achievement of our industry, our country. From India during the independent at that time we are very lacking in the milk production. Now we are in world we are leader in milk production now. Then availability of but even that situation, the livestock production of our country is about 191 million. Out of this, Deshi cow is 151 million, and buffalo production is about buffalo milk. Buffalo population is 108 million. That is 4.5%. Sheep population is 65 million. That is 9%. And goat population is 135 million. That is 4%. And Maharashtra's milk cattle population is 154 lakh population. Out of this, Deshi cow is having the 118 lakh population. And this is a very, that's why conservation and improvement of this breed is very important. And average, we know the average milk yield of the Deshi cow is 3.41 kg per animal, per cattle. And in crossbreed, 7.33 kg per cattle. Buffalo having 4.76. And now our government of Maharashtra has sanctioned the this is a prestigious project for our MPQ Rauri, that is the conservation and improvement of this Deshi cow, the indigenous cattle. This project has sanctioned, and they have provided 2 crores rupees per financial fund for this project. And this is a very prestigious project of the university, and our Vice Chancellor P.G. Patil is looking in this project very seriously, and very they are by open-heartedly they are supporting this project. And that's why this on the decision has taken, I think in the research council meeting, and at that time we decided to make the brainstorming, organize the brainstorming session on this aspect for conservation and improvement of indigenous cattle. And that's why we organized this brainstorming session on this today's today. Object of this conservation and improvement of indigenous cattle is the conservation of superior Shaiwal, Gir, Red Sindhi, Tharparkar, Rathi, and we included the Rap breed also. The one is the Kilar breed in this program also. And to study the comparative production and reproductive performance of the indigenous breed. To study, next object is to study the study the comparative nutrient requirement of this this indigenous cattle. And next object is the comparative quantitative and qualitative studies of the composition of the milk of this Saiwal, Gir, Tharaparkar, and Red Sindhis, and all these Deshi cows. And next object, last object is to establish the biogas plant for energy production and replacement of dung, urine for sustainable farming, and the extension of this program for the scientific breeding, conservation, propagation, and breeding through the awareness campaign. This is the object of this project, and that's why our team, Pune College team, under the leadership of the Somnath Mane, we are doing this project very well, and we want to inputs from this today's brainstorming session, more inputs from the house for this conservation and improvement of the indigenous cattle. And that's why we organize this project with the interest of Honorable Vice Chancellor Dr. P.G. Patil. We organize this program today, and I welcome again. I think Dr. Ulmik, ex-dean, is also attending this program. Sir, welcome on behalf of MPQ Rahuri, sir. And I welcome to all of you and wish you all the best for organizer for this conduct of this program. Definitely, it will help for the concrete solution and concrete suggestion for the conservation and improvement of this indigenous cattle project. Thank you very much.
Thank you, sir. Thank you very much. Thank you for the welcome address to the galaxy of intellectuals. Now we will move towards the technical session. The chairman for the technical session is Dr. Prashant Kumarji Patil, Honorable Vice Chancellor. The co-chairman is Dr. Dinkar Kamble, and rapporteurs are Dr. Sanjay Mandak Male and Dr. Pramod G. Sakre. A round of applause for our first speaker, Dr. Shyam G. Zawar, the most enthusiastic, energetic, and young scientist who is having 47 years of experience and having a very young age of 74 years. A doctorate was confirmed on the subject of entitled Studies on Super Evolution and Embryo to Dr. Shyam G. Zawar. He is BVSc Animal and MBSc in Animal Reproduction, PhD. His PhD thesis is the first doctorate on subject in the country, having 47 years of experience in the field of assisted reproductive technology in sheep, goat, cattle, and buffalo. The first veterinarian in the country to start embryo transfer in sheep and goat. He visited Nigeria in the year 1983 on the invitation of government of Nigeria. He was invited as a guest speaker in 23rd World Veterinary Conference held in Montreal, Canada. He is a member of committee constituted by International Embryo Transfer Society, USA. He was also member of the Central Advisory Committee, Government of India. He worked as a member of group committee for 11th five-year plan of Planning Commission, Government of India. Also member of advisory committee constituted by Planning Commission, Government of India, and on matters related to the development of animal husbandry, dairying in states and union territories. Appreciation for the rural development work by His Excellency, the President of India, Dr. APJ Abdul Kalam. He's a member of many international societies, and he travel extensively in the world for participating for various conferences and seminars. And most importantly, what I touched is he is on the ground and helping so much for our Sahiwal Club of Maharashtra WhatsApp group, and he's sharing so much of views to the farmer and interactive to the farmers. I heartily welcome you, sir, and with a round of applause, and I request you to kindly start your presentation.
Thank you very much. Good morning to all of you, and respected Vice Chancellor Dr. Prashant Kumar Patil and all other dignitaries. First of all, I am very much thankful for giving this opportunity to talk on the subject which is very close to my heart. As rightly said, for the last 46 years, and I always dream of embryos and embryo transfer related issues. I just remembered this morning, because once you do a doctorate thesis, normally you forget to refer back the thesis again. But when I referred my thesis this morning, you know, I have great memory of the Mahatma Phule Krishi Vidyapit, wherein in 1983, that time the Salunke Vice Chancellor, he invited me to do some work on embryo transfer in goat, and which was your Angora goats over there for the mohair production. So my thesis has got a contribution even from the goat work which I have done with the Mahatma Phule Krishi Vidyapit. So that's really, I feel very proud to be back again with all the dignitaries of the MPKV. Now I will straightway start my presentation and about the work which we have done it. So I'll go ahead and share my presentation. So let me. Just give me a minute.
May I help you, sir?
No, I think I should be able to find out. Just give me a minute,
Fine,
please.
sir.
Don't mind.
Fine, sir. Fine, sir.
Yeah. Is it visible?
No, sir, not yet. Not
Okay, I
yet.
need to share screen has been already shared for me?
No, sir. Just back to the Zoom and click on share screen.
I'll do that in a
Yeah.
minute. I'll do it in a minute.
So click on the share screen button.
Yeah, I'm not able to just see that button. It's just I've been doing so many times, but
Are you able to see the down arrow, the down arrow there, or a small window?
yeah, just gives 1 minute. Yeah. Okay, got it. Yeah.
Yeah.
Is
Yeah.
it
Yeah, perfect.
okay?
Yeah.
Thank you.
I'll just
Go
make
back
it
to
full
my
screen.
first idea.
Yeah.
So, so thanks again. As the topic which has been given to me is the role of in vitro fertilization in conservation of the indigenous cattle. It's not moving.
So click on the slide. Click on the slide.
Okay. Okay.
Right.
Yeah, okay. Let's look at the different ways of the genetic improvement in the cattle, four ways which are there. One is the natural breeding, which everybody knows about it, but which is very costly to maintain a bull of your choice and which makes a very less choice for the genetic variation. That's the standard practice which has been used for years together. Then came the artificial insemination, which is quite cost effective, but again, it taps the only male genetic potential, and but it takes longer time to achieve the desired genetic level. And the third technology which came was multiple ovulation and embryo transfer. This was somewhere around in the year 1980 onwards, which was there almost for about 2 decades. Various agencies worked on this. This is known as multiple ovulation and embryo transfer, which is a little expensive process, and it involves all the reproductive tract of the donor cow, and the donor has to be in a good reproductive health. These are the basic prerequisite for doing MOET. When we come about the last and the latest technology, which is called as ovum pickup or in vitro embryo production, IVF, exactly like test tube baby in human beings. And this is one of the most advanced breeding technique. No doubt it's expensive at this moment, but it has got a multiple advantages, which you will see during my presentation. It taps both the male and female genetic potential up to the fullest potential. And in case of this whole process, we are only using the ovary. I hope I am audible to everybody clearly.
Yes, sir. Perfectly audible.
Thank you. So let me tell you a quick difference between MOET and what is the difference between IVF. In MOET, you can see this uterus, we are showing it, wherein the multiple ovulations are done by giving a stimulation by injecting hormones. With that, we do a multiple ovulation on the ovaries. And then we do artificial insemination with a good semen, and the embryos are flushed out from the uterus outside. These are the 7-day-old embryos which are made into the uterus. That's why it is known as in vivo. And exactly opposite to that is the in vitro technique. In vitro, what we are doing is that from the ovaries, we are taking out the oocytes, which are immature oocytes, and which you mature them into the lab over a period of 7 days, produce embryos, and then you transfer them. So rather you create an artificial uterus into the lab, which in vivo, in MOET, is all done by the uterus itself. So there is quite a big difference between MOET and this IVF process, what we call OPU, ovum pickup. But it has got a tremendous advantages now all over the world. This IVF has taken over the MOET, and millions of calves are being produced in Brazil and United States. I had an experience of working with both the things, especially when I started my career well back in 1975, when I started my work with the sheep at the Raymond Sheep Breeding Farm. It was all surgical process, and even when I did the work with the Angora goats at Rahuri, MPKV, that time also it was all surgical intervention. I mean, everything was embryos were collected surgically, and then they were transferred also surgically. Even in the year 1977, I remember that even the process in cattle was also surgical. We were collecting the embryos surgically and even transferring it surgically. The invention of the non-surgical collection of the embryos through MOET and transfer non-surgically, really it made a big difference. And further, again, with the IVF, it further strengthened the whole thing. Now, these are some of the major IVF lab equipments which are there. As you know, I was there almost for about 5-6 years, even managing as a CEO of this laboratory near Vadgaon, in Vadgaon Rasai in Taluka Shirur district, Pune. So these are the equipments which are there in that lab. I just retired from that organization only about 2 months back. And these are the equipments, very high tech equipments, first microscope and the embryo freezing machine. These are another interesting equipment, benchtop incubator. This is nothing but acting as a uterus into the laboratory. You know, same equipment are being used for the human beings, no difference. So I just will go through this quickly through those equipments. I mean, nothing much into these equipments. These are the equipments which are required for collection of the oocytes. Now, let me explain you a little bit about the reproductive tract of a cow. This is how a reproductive tract of the cow, it looks like. You start with this vagina and vulva and the vagina, and then you have a cervix, mouth of the cervix, and then it is divided into 2 horns, which we call as the left horn and the right horn of the uterus. And then it's with the fallopian tube. The uterine horn ends with a fallopian tube, and then there is an ovary, from where actually the ova are being released. In a regular cycle, which in cows is around 21 days, you will have one ovum which is being released. And then if the insemination is done at the right time, the semen will travel, and only one lucky sperm will get into that egg, mature egg, and the fertilization will occur and a pregnancy will occur. Now, in case of MOET, as I explained you, the ovaries are overstimulated. So you may get about 5 to 8 eggs released from the ovary, and they have to be fertilized with the semen. So, and after 7 days, when they are embryonic stage, you have to flush them away, flush out from the uterus, both from the left horn and the right horn. That's how this MOET process is done. Now, in case of this OPU, which is a transvaginal oocyte recovery, wherein what we do is that one hand is put into the rectum, and then there is a needle holder, OPU needle holder, what we call, through which there is a needle attached, and you catch the ovary through the rectum, and then it is punctured, the ovaries, you point out the follicles with the help of an ultrasound. So you have to do a multifarious actions at a time, one person, even holding the ovary, then putting the needle into the follicle and rupturing it and sucking out through a suction pump these immature oocytes, what we call, they will be all immature oocytes on the ovary. I'm just referring you a point here. You know, in case of bulls, we collect the semen and we spread and use it over thousands of animals, and even about 1 million, about 0.1 million doses also are made from a bull in a year, in a lifetime. As against, female, when it is born, a female calf, when it is born, it has got a tremendous capacity, but due to the physical limitations, you can't have so many babies produced out of it. Only one embryo develops, and one calf, sometimes you may get twin. So in a lifetime, a cow may give at least about 8 to 10 calves, whereas her ovary has got a capacity to produce about 50 to 60,000 oocytes. But theoretically, it's okay, but practically it is not possible. But with the intervention of IVF technology, there are records wherein up to 200 to 300 calves from one single cow, one single donor cow, have been produced in her lifetime. We ourselves have got a few examples, which I will show you further into my presentation. Now, this is in short a process of the collection of the oocytes. So you do it for the left ovary first, and then the right ovary, another one. So these are the different equipments, which are used. This is a very critical equipment into the whole process. A yellow color, it's an ultrasound. And this is a suction pump. And the next one is, this is the probe, needle holder. The tip of it, which is echogenic, which is put across the ovary. So it catches the images and you can see those images on the ultrasound screen. And this is the needle which you have to use it and you have to bring it into the follicle. A follicle on the ovary, there are different type of follicles are there at different stages. There are small follicles which can be varying from 3 millimeter diameter to even up to 10 millimeter diameter. So every follicle should have a oocyte into it. That's an immature oocyte. And one good thing of our indigenous cattle, this Bos indicus breed, is that you don't need to do any stimulation. They are prolific oocyte producers. That's the best part of it. You know, you don't need to use any hormone, and you just take a cow and then you start aspirating it, the immature oocytes. These are the whole setup of the equipments. If given an opportunity, even right now also I enjoy doing OP and all that, because that's how I really enjoy the whole thing. Now, this is a interesting video you can see. This is the ovary, which you are seeing an ultrasound image on that. And these all the black spots represent a follicle. So a needle will come from the top, which is, we are putting it through the cervix. We are just holding the ovaries and then putting the needle from the top into the follicle, and with a suction pump with the paddle at the foot, we aspirate it. So this is a few seconds video you can see. This is you'll be able to see a needle coming from the top. You can see this. And once it sucks, that black, because it is, as it has got a cavity in ultrasound, the image is black. So once it gets punctured and you suck it, it will again, like it's a blister, it will collapse. So every follicle of this should have a one oocyte. That's we work out as a recovery rate. Normally, you don't get all the oocytes which of the follicles you have punctured it, but even if you get about 50 to 60%, it is good enough. So this process, we do it on one ovary first, and then we do on the second ovary. So every small follicle on the surface of the ovary, we puncture it and aspirate those oocytes. Now, once this is collected into a tube, this is a follicular fluid in which the oocytes will be there. You filter it. I mean, there is a special filter which all extra fluid will come out of it, and only the oocytes will not be able to pass through that filter. Now, there are three. I don't want to go into much of the technical details of the whole thing, but these are the three important processes which are required: in vitro maturation, in vitro fertilization, and in vitro culture. Why we need an in vitro maturation? As I told you, we are collecting the oocytes which are not mature. So they need to be matured into the laboratory by putting them into the right media and giving them all the necessary nutrients and other things which they would have got into the uterus. So this is the first step, IVM. So once you collect those oocytes from that filter, you examine them into detail under the stereo microscope, and then these oocytes are put into these oocytes are graded. See, this is a oocyte, one oocyte, this is one oocyte, these are the A grade oocytes. How do you differentiate A, B, C, D? A very quick differentiation is that these are the cells which are around these immature oocytes. They are known as cumulus cells. More the number of layers you have got, thick layer, they are classified as A grade. Then this is less layer, B, and C further less, and D, and these are E. So almost you don't make embryos out of D and E normally. So A and B and C will result into the formation of embryo. That's how we classify these oocytes. Now these are the immature oocytes, selected oocytes which are being put into the incubator, and they are put about for 23 hours. And on second day, you see these oocytes again, and you can see the cells around these oocytes have quite loosened. So these are the mature oocytes, and now they are ready for the fertilization. If you put a sperms here, they will not get fertilized. So you need to mature them. And for all this purpose, we have to use the medias. And these media, unfortunately, we have to import it from the other countries. IVM media, IVF media, IVC media, different medias are imported from Brazil and Denmark and a couple of other countries. So post-maturation, these oocytes now are ready for doing the fertilization. Now, I'm showing you a micro-Q incubator. These are the incubators which are maintained at a particular temperature, 37.5 degrees centigrade, but they don't have any gas supply. But if you want to, there is a particular type of media wherein you can bring in the oocytes. You can collect these oocytes somewhere in maybe even Jammu and Kashmir, or you can bring it to your lab in Pune, as long as they reach in within 24 hours to your lab. So these incubators we have been using when I was in JK Trust. We have been using all across the country for transporting the oocytes from one end to another end. Now, we have to do a lot of preparation of the semen, which I will not go into the details, but quickly, all detailed examination of the semen which we have to use it. Preferably nowadays, we are using sex-sorted semen as we are more interested in getting the female. So we can produce 90% of the female. So all the semen which we centrifuge it through different steps, study it, and ultimately we look for the semen pellet. You can see into the third tube here, which will have all the live sperms. And then a count is done, and then we decide as to how many sperms we have to put it with the number of oocytes. There is a ratio, and accordingly, these sperms, the washed pelleted semen, is put with the oocyte. So this is a very good small video. You can see these are 1, 2, and 3 oocytes are here, and these are the sperms. You will see they are going around the oocytes, and even oocytes are encircling them. They are encircling them, and only one lucky sperm gets into it, and then there is a chemotaxis reaction, and then there is no further entry. No entry is there for the future sperms. So you can see these sperms moving around the oocytes, trying to get an entry for doing the fertilization. So this is how it all depends on the quality of the semen, the type of bull which you are using it, to know how you get the best fertilization rate. So now after that, these oocytes along with the sperms are kept for another 16 to 18 hours. And on the third day, you see these structures which are known as a potential zygotes or presumptive zygotes. We get some indication in some of them that they have done the fertilization. Fertilization has occurred into that. But really, you don't get a very clear picture as to how many embryos you may get out of it. So this is the third day. You examine them. All the cells around them, whichever that cumulus cells, if they are which were there earlier, they are being stripped away. You use a stripper and then you take them out to these surrounding cells. So these are the clear-cut potential zygotes. You put them again into under the benchtop incubator, which I showed you some time back. This is the one which has to be maintained at 38.5 degrees centigrade. And this has got a tri-gas supply of oxygen, nitrogen, and carbon dioxide connected with the cylinder. So that has to go uninterrupted, and uninterrupted electrical supply also has to be there, because this is where these potential zygotes will remain for another 5 days till you see them as a well-grown embryos. Now, quickly, as many of you will know, we start with a 1 cell, then it will get after fertilization 2 cell, and multiplication occurs like this: 4 cell, 8 cell, 16 cell, early morula. When I was working with a sheep, normally we were sheep and goat, you collect the embryos which are 16 cell embryos, early or a morula. That's how. But in case of cattle, when you are collecting them, when you are examining them at 7th day, the picture is different. These are 6.5 to 7.5 day IVF embryos, which are excellent. This is again a compact morula. Next stage is early blastocyst and expanded blastocyst. So in case of IVF in cattle, when we do it, normally you land with the blastocyst or expanded blastocyst, and these are the one which are ready for transfer. Now, their choices are there for the transfer. you can do a fresh transfer, or if you don't have sufficient recipients in which the embryos are to be transferred, then you can use freezing machine and freeze them. We have to keep it in mind that when we are freezing the semen, in every straw of the semen, there are 20 million sperms. In conventional semen straw and in sex-sorted semen straw, there are 2 million sperms. But when we are freezing the embryo, it's one single embryo which is put into the straw. So you can imagine the difference between the freezing of semen and the freezing of embryos as well. This is a cluster of embryos. There are 43 embryos, which was a record in the country, which we got it by collecting 103 oocytes from a Gir cow. And we got 43 embryos out of it. Really, I mean, she's a phenomenal cow. I'll talk about the same latter. Now, just for your interest, I mean, it's changing my subject and little bit change from our regular talk. A quick, nice video I thought I'll share with you. You know, this is a video time lapse with the human embryos. This is, you can see the date at the back, at the bottom. And in this date, it is, see, 22nd of October 2008. And every second, the camera is being feeded, and how the fertilization of the human embryos are occurring. They're all numbered, you know, they have been given alphabet. You can see the second wise how the photography is occurring. Which is known as a video, a time lapse, which is very interesting. Have a look at it. It's, I think, few seconds. You can see the date changing down below and how the time clock is changing second. And you can see how the division is occurring into the embryos, 2 cells, 4 cells. And, you know, these are realistic. I mean, it's not a artificial, but it's a realistic video. I got it in one of my trips from USA. So always I present it to, I always like presenting this. And you will see how they expand it, and they will come out of that zona pellucida, which we call as a hatch blastocyst. You will see gradually each one. That's why they have been identified with these alphabets. So they will break the shell, and then they will come out, which ultimately gets attached to the internal wall of the uterus, to the endometrium. That's how the pregnancy further develops it. So we were there on, I think, 22nd of October. Now it's 25th of October, the time. It's because in human being process is about 4-5 days. You can see this H really expanding it, and it will now just come out of it and hatch it. See, this one is also expanding. So each one of these embryo is being shown how it hatches and it lives. You can see this embryo which is coming out of the zona. So I just, sorry, I changed my topic for a minute. Now we summarize the flow diagram of OPU. We take a good donor cow, then we do this ovum pickup. We collect the oocytes, we fertilize them, and these embryos are cultured, and then these embryos are either transferred into the recipients or you can freeze them. Now here I would like to tell you how, in relation to our conservation of indigenous animals, it can play a dramatic role. Here I really want to mention the efforts put in by Dr. Mane, to whom I know for years now. He's really so keen, being a president of the Saiwal Club, and always actively involved and for the conservation of the indigenous breed. Now, these indigenous cattle particularly, and as a Rashtriya Gokul Mission, which is a mission of the government of India, wherein, you know, there are 31 IVF labs which are funded by the Ministry of Animal Husbandry, Government of India, and I am as a national mentor for that. And also we have a CMU, Central Monitoring Unit, recently made a Central Core Committee for IVF activities in the country, wherein I am heading as a chairman with another 5 members, colleagues of mine. So this indigenous animal at our farm at Vadgaon, in the JK Trust farm, we worked on different breeds: Gir, Saiwal, Tharparkar, Rati, Red Sindhi, Angol, and even Pungunur embryos, we got it from some place. So we worked on different breeds, and one good thing which we started about 2 years back is that we had some very high milk yielding donors at this JK Trust farm. And from Brazil, JK Trust was the first organization who were allowed to import the sex-sorted semen of the Gir. And such embryos which were produced, they were transferred into the recipient, in the crossbred recipients of the farmers of the Ahmednagar, Pune, and Satara district, wherein it was very interesting scheme. Some people know, some farmers, they are not interested in rearing the calves. So we thought of that idea, keeping in mind the Anand pattern which is there in Gujarat, wherein the wombs of the ladies are hired for the human pregnancies. Taking that as an idea, we started hiring the wombs of the cows, crossbred cows, and we started paying to the farmers rupees 10,000, 5,000 at the 5th month pregnancy and 5,000 at the delivery of the calf. And then this calf was brought after 4 days, when it was 4 days old after the birth, and was reared at our farm. So this was another good way of increasing the genetically superior animals using crossbreds recipients. This is an interesting incubator for transporting embryos across the country. This was we were always taking as a hand baggage, and you can put about 50, 60 embryos into this, and they don't need any air ticket. And these are the passengers traveling by air without any air ticket. Now, this is how the embryos are there. This is another micro-Q incubator where you can load one embryo into each straw, and then you can take this as a micro-Q incubator. Now, let me tell you what is the effect of this IVF technology and how can we utilize this for conservation of our indigenous breed. It can be applied in a big way. And as I told you, there are about 31 labs which are funded under Rashtriya Gokul Mission by Government of India, and there are about 8 private labs which are working in the country. So all these near about 40 IVF labs come under the purview of our this central monitoring unit, which is recently made by Government of India. Now, this is a premium cow. Many people must have heard about this Gauri, which is still there at the JK Trust farm. And from this cow, we have produced almost 135 pregnancies have been established. So she has almost produced up till now more than about 125 calves just in a period of 2 years. Imagine. So these are all her calves. Her calves are being spread right all across the country, right up to Kerala, because the embryos of her cow, this cow has gone to many places. So she has become a very famous cow, a Gauri, a premium cow. And these are the different embryos which were collected at different stages from this cow. These are the Gauri female, male, few calves are here. Now, another cow which joined this is Samadhi. It was named as a Samadhi. We got it from one of the Gujarat temple. And she made a record of 103 oocytes, and I just now told you 42 embryos. And from her also, almost 100 pregnancies are being touched. So this way, very elite cows, we can explore them, and the one which are really doing good, once they produce good number of oocytes and good number of embryos, they will be always a good producer. And such animals can be, their embryos can be put into the crossbred cows, and that's how you can multiply any breed. I'm talking about Gir, but we have done on Sahiwal, Ongol, all breeds. And best advantages, again, I'm repeatedly telling, for indigenous animals, they produce very good number of embryos. Now, quickly, this is, we are talking about cattle, but I wanted to mention that IVF in cattle was introduced in 2016 by JK Trust in a big way, and about thousands of IVF cows were produced by them over last 5-6 years. We also succeeded doing IVF in buffaloes at one of the large buffalo farm near Pune, Sorone farm. So country's first IVF cows were also produced there. IVF twins are also produced. And you must have heard a big story about a Gir district, which is known as a Gir Somnath district in Gujarat, where these are the calves produced a bunny breed, which is a very tough breed of that Kutch region. And our Honorable Prime Minister was talking about the same breed a day earlier when we were doing about this work. It was just a sheer coincidence. So these are the 3 bunny calves from one single buffalo calf. And to conclude my talk, at the JK Trust, we had a privilege of visit of the Secretary Animal Husbandry, Government of India, as well as the Union Minister for Animal Husbandry, Mr. Purushottamji Rupala, who also came recently, wherein even he had an interaction with the Dr. Somnath Mane also. And I should say that the amount of work which is being taken up by the MPKV, Rahul, is also commendable. And if anything, we can be of help under Rashtriya Gokul Mission, it will be our pleasure. If a project proposal can be prepared and it can be submitted, or it can be, routed, I will try my best to get some funds for this project, because you are doing a great job. And I don't want to, I think I've already taken more time, but I will rather love to answer any questions, because as you know, I'm so committed with my subject, and I love my subject definitely. So thank you very much once again for giving me this opportunity to make this presentation to all of you. Thanks.
Thank you, sir. Thank you very much. By listening to your presentation, I recall the quote of Gulzar, who's mentioned about Salil Chaudhary. He mentioned that the kind of intoxication and energy that bursts forth when you shake and uncork a bottle of champagne. The same thing I want to recite. Full of life force, mesmerize and charm in your presentation. Thank you, sir. Thank you for being with us. Now I like to invite next speaker, Dr. Rakesh Kumar Pundir, sir, is currently working as a principal scientist in Animal Genetic Resource Division, National Bureau of Animal Genetic Resource, NBAGR, Karnal, Haryana. He did his post-graduation and PhD from IVRI, Izzat Nagar, Bareilly. Presently, he is engaged in research on characterization, evaluation, documentation, and conservation of indigenous cattle and buffalo genetic resource of India. He had more than 30 years of research experience and completed 33 research projects in the field. Breed descriptor were developed for 22 cattle and buffalo breeds population. From his work, 9 cattle breeds were registered as new distinct breeds. He has published 17 breeds monograph, 4 books, 2 training manuals, 97 research paper. He acted as a general secretary for Society for Conservation of Domestic Animal Biodiversity for 5 terms, each of 2 years. He also acted as a member of different advisory committees like Board of Management, NDRI Karnal, Institute Management Committee, PDC Merit, Editorial Board, Journal of Livestock Biodiversity, Secretary QRT, NBHR Karnal, and Nodal Officer, Cattle Genetic Resource ICR. He has organized different national symposium, brainstorming session as well. And proud to tell you that he is a fellow of NADS, SOCDAB, ISCA, and BioVed. I heartily welcome you, sir, and I urge you to roll your presentation.
Whether my slides are visible?
Yes, sir. Your slides are visible.
Honorable Vice Chancellor, Dr. Patil, Deans and Directors of the University, Head of Division, faculty members, and participants. I am supposed to speak on indigenous cattle and their conservation. I am from National Bureau of Animal Genetic Resources, Karnal, that was established in 21st September 1984 in Bangalore. Later on in 1985, it was shifted to Karnal. And in 1995, actually there were twin institutes. One is the National Bureau of Animal Genetic Resources, another is the National Institute of Animal Genetics. Both were merged and renamed as National Bureau of Animal Genetic Resources, Karnal. Our mandate is identification, evaluation, characterization, conservation, and utilization of livestock and poultry genetic resources. And recently, policy planning is also attached. Our major programs are identification and characterization of livestock genetic resources, livestock genomic studies, conservation of farm animal genetic resources, registration and documentation of livestock genetic resources, and sustainable utilization of livestock genetic resources. If we look at the strength of our cattle, no, this is just wait.
So you can use
Just-
the arrow keys.
Just wait. I want to keep it in some other place. Animal genetic resources are the species or animals. Those can be used for food in agriculture. They are the most valuable and precious, and they are strategically important asset of any country. They are the raw material for the breeders for improvement and enhancement of productivity, and they can be represented in terms of breed, strains, and different populations. Actually, this bar is disturbing me.
Sir, you can use the arrow keys to go forward. Right
I
out.
I want to remove this one.
Are you are you facing the issue with the pop-ups?
Yes.
Okay. Can you please click on the escape?
Yes.
Or else you can share it again. Close the sharing. I'm closing your sharing. I share it again, sir. Yeah. Now click on the full screen. Just above. Make it full screen, sir.
I'm not able to look. Okay, just wait.
PPT mode.
Perfect. Right. Okay. Now, can you please check with the arrow keys from your laptop keyboard?
Okay.
Fine.
How we define the breed? Breed is a group of domestic animals. Those are having homogeneous appearance, phenotype, and behavior. Characters that distinguish it from other organisms of the same species. They are formed by the genetic isolation, either by natural adaptation or selective breeding or combination of both. The breeders establish a breed by selecting individual animals from within a gene pool that they see as having the necessary qualities needed to enhance the breed model they are aiming for. These animals are referred as a breed foundation or a breed origination. Breed have the specific characteristics, also known as the breed traits, and they are inherited in purebreds from generation to generation. Domestic animal breeds commonly differ from country to country and from nation to nation. Breeds originating in a certain country known as native breeds or local breeds. If we look at the strength of our cattle in our country, we are having the largest population of buffalo in the world and second largest cattle population. We have large biodiversity as we are having 50 breeds, registered breeds. Those are having in shape, size, color, production, productivity. So almost we have very high biodiversity in our cattle populations. They can survive in extreme of the agroclimatic conditions. There are breeds those are easily surviving in the cold climate like Jammu and Kashmir, and another area like hot climate like Tamil Nadu and Karnataka, Maharashtra. They are well adapted to high temperature. They have powerful drop capacity and resistant to tick and disease. We have world best buffalo breed, that is Murrah, and Ongole, Gir, Sahiwal breeds were imported by several countries for increasing resistance and disease and heat tolerance. But they are better in under extensive system of management and low of the productivity. If we look the cattle population or livestock population in our country, as per the 2019 livestock census, we are having around 192.45 million cattle, 109.85 million buffalo, 74 million sheep, 148 million goat, and around 9 million pig. If you look at the composition, we are having 36% cattle, 20% buffalo, 14% sheep, 28% goat, and around 2% pig. If you look at the bovine population trends in our country, the first livestock census was reported in 1951, and there were 155 million cattle. These cattle population were increased and reached at the maximum 204 million in the year 1992, and they were declined. The first time crossbreds were reported in 2008, and our cattle population, after getting the crossbred population, it increased from 8.5 million to 50.42 million, while indigenous cattle declined to 142 million, while our buffalo population increased significantly over all the years since 1951. If we look there, crossbred, exotic, and indigenous animals were declined from 150 million to 142 million from 2012 to 2019, while female increased. Males again declined. In case of exotic or crossbred, they were increased from 39 million to 50 million. Female increased, males declined, and total cattle increased from 199 million to 192 million, a very marginal increase. If we look at the bovine population in Maharashtra, in 1982, there were 16.6 million cattle. In 2019, it declined around 13.9 million cattle were there. The buffalo increased from 3.97 to 5.6 million. If you look at the exotic crossbred indigenous animal in Maharashtra, non-indigenous, including non-descript, again declined. Females again declines, males decline. Crossbred increase. In crossbred, females increase. In crossbred, males decline. And crossbred total cattle decline from 15.48 million to 13.99 million. We are having 50 registered breeds in our country, and among them, 87 are the local breeds. The breeds, those are originating in the single country. So around 87% the breeds, those are only available in our country, like Burger, Khillar. There other category of the breed is the regional transboundary breeds. The breeds, those are available in different countries, but within the region, like in Asia. Siri is available in India and Nepal, Thar Parkan in India, Pakistan, Red Sindhi in India and Pakistan. Another category is international transboundary breeds. The breeds, those are available in different regions like Asia and Europe. So Sahiwal is available in many regions, Gir, Ongole, and Nellore. The breed-wise census was first reported in India in the year 2007, and they reported 44 breeds or population. That was updated in 2013, and they reported 37 breeds. Those were registered at that time. This is the distribution of the breeds in our country. If you look at the utility pattern, we can divide all the cattle breeds in 4 milch breeds like Sahiwal, Red Sindhi, Gir, Thar, Parker, Rathi. For dual purpose breeds, those can be used for draft and as well as for the milk. Haryana, Kankrej, Dioni, Krishna Valley, Ganga Tiri, Binjarpuri, Mewati, Malvi. For draft breeds, those are used for the agriculture work like Hallikar, Kangayam, Khillar, Ongole, Amrit Mahal, Nagori, Red Kandhari, Nimari. So most of the Maharashtra breed, except Dioni, they are coming in the draft category. If you look at the production system in our country, we have intensive production system. Very few animals are there in that system. Stall feeding is there, good facility for management is there, AI is there, marketing of milk is there for public and private enterprise, and proper recording system is there. But in this system, very few animals are there. Another system, the semi-intensive or crop livestock production system. This system is majorly available in our country, in which few cows, bullocks and followers are kept by the farmers. Productivity is low to medium. They are kept on grazing with a small amount of stall feeding. Majority of the indigenous cattle are non-descript animals. Majority of the animals are under natural mating, very few in AI. Milk is partially consumed by the farmer's family and partially sold. Bullock sold on good price and dung used as fuel manures. Another system is the pastoral system or extensive system of management, where big herds of cattle are maintained and kept on migration. The migratory herds of Kankrej, Nari, and Pullikulam are very common in this system. They are the few important breeds like Sahiwal. This is a best milk breed in our country, available in Punjab, Rajasthan, and Haryana. The breed is utilized for improvement of local breeds for milk production, and they introduced in different 17 countries. Most of the animals are under extensive system of management. Heavy and symmetrical body is there. Animals are lethargic. Teeth are long and cylindrical shape. If you look the population in 2013, there were 10.9 lakhs cattle purebred, 37.8 lakhs graded, and total 48.8 lakhs in our country, including grades and purebred, and they are representing around 3.23% of our cattle population. Distinguished characters: reddish dun, pale red or brown color, heavy breed but lethargic, symmetrical body and loose skin, horns are short and stumpy. Another breed is the Red Sindhi. They are available only in the organized farm, and very few animals are there. Originated from Las Bela cattle of Bluchichan, a dairy breed. Other is Capsius and Pendulous. No animal available in field in our country. There were around 59,000 animals were there, and total, including grades, they are representing around 0.37% of cattle population. Red in color and underbody parts showing whitish gray color. Ears were moderate in length and dropping. Eyes were black and shrinking. Head is well proportionate with an occasional bulge on the forehead. Horns were small to medium in length and curve. Another milch breed is Gir breed that is available in Gujarat and Rajasthan. World-renowned breed for its tolerance to the stress condition, resistance to various tropical diseases, reared by professional breeders like Rabris, Bharwadi, Maldharis, and Ahir. Imported to Brazil, US, different countries. Animals lethargic or pendulous, sheath, and heavy build. Hardy animals can produce in different environment. They are representing around 3.38% cattle population, including purebreds and grades. The distinguished character is the forehead broad and convex like a bony shield, long and pendulous ears like a folded leaf, and peculiarly curved horns. Another milch breed is the Tharparkar breed. Those are available in Rajasthan and well adapted to hot climate of this area. Named after Thar desert, well adapted to desert condition, adapted to scarcity of fodder and water. During summer, survives on small bushes, well developed udder. They are representing around 0.48% of cattle population, and distinguished characters are white or light gray body color. Forehead is broad and flat or slightly convex. Develop each loose and flexible but not voluminous. Face is lean, fine, and slightly ditched to muzzle. Eyes are full and placed. Another milch breed is Rathi breed that is available in Rajasthan. They have symmetrical body. Develop is voluminous. Milch breed named after from pastoral tribe Rath. Originated from mixture of Sahiwal, Red Sindhi, and Tharparkar. Thar desert is scarcity for fodder and water. Animals are docile. They are representing around 0.82% of the cattle population, and distinguished characters are symmetrical body, brown with white patches of all over the body, sometimes animals having completely brown or black coats with white patches. Develop is voluminous. Face is broad between eyes and slightly ditched. If you look the cattle breeds of Maharashtra, we are having around 6 breeds of cattle. Dangi, Dioni, Gelo, Khillar, Red Kandari, and Konkan Kapila registered recently as a distinct breed from the Maharashtra. Dangi breed is declining from 2007 to 2013 and available in Nasik and Ahmednagar. They are reared for the drought purpose and poor milker. Coat color is white with red or black spots, medium in size with deep body. Head is usually small with slightly protruding forehead. Muzzle is larger. Horns are short and thick. Dollop is slightly pendulous. Hooves are black. Skin excludes an oily skin, white protect from heavy rain. This is the typical character of the breed. Then Deoni is also declined from 1.65 lakh to 1.19 lakhs. Available in Latun and Red Parbani and Usmanabad. This is dual purpose breed. Body color usually spotted black and white. And these breeds have different 3 strains: Blanca, Vanara, and Severa. Ears are gray, white, or complete white with black pinna. Next is the Gallo breed. That was also declined from 2.22 lakhs to 1.21 lakhs. White, this data is from 2007 to 2013, and after 2013, there was no live breed census available. They are white or light gray color. Males are generally gray over the neck, hump, and quarters. Medium in size, light built, narrow and long body. Head is long and narrow, usually tapering towards the muzzle. Eyes are almond shaped. Ears are medium in size. Horns are short and stumpy. And Khillar is also declined from 1.41 lakhs to 1.10 lakhs. They are available in Satara, Kolhapur, and Sangli. Drought, used for drought, poor milker. Grayish white color. Males are dark over the forequarter and hindquarters. Compact and tight skin, long and narrow forehead. Horns long and pointed. Red Kandhari, this is the breed which increased over this period from 2007 to 2013. It increased from 1.76 lakh to 2.35 lakhs, used for draft and poor milker, available in Bidar, Latur, Parbhani, and Usmanabad area. Animals are medium in size, strong and robust animals. Body is compact and deep dark red color. Forehead is broad between eyes and slightly bulging. Horns are evenly curved. Ears are long and dropping, and this is typical characteristic. Eyes are shining with black color around the eyes. Horns are thick at the base and tempered to the blunt point. Another recently registered breed is Konkan Kapila, that is from the Konkan region of Maharashtra and also available in Goa. Reddish brown or black gray mixed color is there. Straight face, small to medium hump and roll-up. We are having process of registration of new germplasm. By the 2008, we have given the SSN number to all the available breed, and that was around 130 breeds in 2018. After that, we have started the process of registering new breeds, and guidelines, descriptor, and application for registration of new breeds are available at NBGR site, as well as guidelines for registration of variety strains. They are also available, and we have registered since then 2008 72 new breeds, and the total tally is 2002 breeds of different species. In cattle we have 50 breeds, buffalo we have 19 breeds, and recently 2 breeds of buffalo and 3 breeds of dog were registered. We are giving the SSN number to all the registered breeds. This is a alphanumeric code. First is country name, India, then species name, like cattle, then A, B, C, D. They are the 2 major states where these breeds are available. Then breed name, then code for the species, and then number for the breeds. A specific number is given to a breed. Like for Motu, we have given the 31. Now come to conservation. Actually, conservation is the sustainable development is called the conservation, and that can be made through genetic improvement programs. The development that meets environment, social and economic needs of present without compromising the ability of future generation to meet their own needs is called the conservation. We have different categories for the conservation. Like extinct breed means no likelihood of reestablishing the population. This breed is gone. And no purebred males and females can be found or available. Another one is the critical, that is close to extinction, and genetic variability is diminished to less than that of ancestral population. It means when we apply the selective breeding, the genetic variability is reduced. Action to enhance population size is essential and survival and propagation. Breeding females are less than 100, then we say it a critical. Endangered. Effective population size is too small to prevent the genetic loss. Inbreeding is there. Due to small size, high inbreeding occurs and genetic variability is reduced. Need to initiate conservation action, and in this category, breeding females are from 1,000 to 1,000. Then vulnerable. Some unfavorable effects endanger to existence of the population, like crossbreeding. Preliminary measures need to be undertaken for checking further decline in number, and breeding females in this category range from 1,000 to 5,000. Then insecure, population numbers are decreasing rapidly. Breeding females are in between 5,000 to 10,000. And if a breed have breeding females more than 10,000, we are saying is a normal. Population is not in risk of extinction and can reproduce without genetic erosion. No visible changes are seen in population size. What are the criteria that a breed need conservation? One is the number, population. We have two breed census in 2007 and 2013. Based on this number, we can say these breed need conservation. Then fast decline. Suppose a breed is having good number of animals, but within the 5 years or 10 years, it's declined to like series breeds. It declined very fast. So based on that, we are also saying that breeds also need the conservation. A breed, if have a unique trait, like Sahiwal cattle is there, this is the best breed of our country for the milk production. So that also need conservation. Then genetic variability. Genetic variability means we have 50 breeds of cattle, but some are the shorter, some are the longer, body color pattern. Then we have the genetic studies, how the breeds are contributing to the genetic variability. Based on that also we decide that breed need conservation. Then economic importance. If a breed is economically important, like Murrah breed, then again we need the conservation. Conservation means it also includes the genetic improvement. If we look 2007 and 2013, number of different breeds. They are the breeds, those are having less than 5,000. They are the Vechur, Panganur, Pulliculam, and Krishna Valley. They all need the conservation. Then we have the breeds, those are between 10,000 to 20,000, like Siri, Burger, Mewati, and Pawar. But if we look the Siri, it was declined from 61,000 to 12,000. A very sharp decline is there. So this breed is need conservation. Like Burger, 20 to 14,000. Mewati, 75 to 14,000. Pawar, 24,000 to 20,000. So if the breed is declining very fast, we need conservation. Amla Cherry, it declined from 2.17 to 13 lakhs to 39,000. It means the breed is need conservation. So based on the breeding females, what are the breeds we need to be conserved? If the females are less than 6,000, they are the Burger, Krishna Valley, Pulliculam, Pangano, Siri, and Vechur. These breeds are need conservation. Another criteria is based on the breeding males and inbreeding coefficient. If males are less than 35, then we need for Mewati, Pulliculam, and Vechur. And based on the inbreeding coefficient, like Mewati, Pulliculam, Vechur, again need the conservation. Then another criteria is based on the sharp decline, like Red Sindhi, Kangayam, Siri, and Amla Cherry. The cattle breeds based on the FAO criteria, like critical, we say Vechur is critical. Endangered breeds, Krishna Valley, Mewati, Pulliculam, and Pangano. And vulnerable is Burger and Siri. For conservation, there are two methods. One is the in situ, another one is the ex situ. In situ, we are having farmers and flocks and different farms, breed farms, organized herds in the breeding tract. Ex situ conservation, the cryopreservation of germplasm like ova, embryo, and semen, and organized farms outside the breeding tract is also come under ex situ conservation. So in case of in situ conservation, the existing organized farms can be act as a nucleus herd, and each breed should have 1 or 2 nucleus farm, strength ranging 2,000 to 250, and these nucleus farm will operate as center of conservation. Then we need detailed survey need to be carried out in breeding tract to identify true to breed animals under field situations, which could be preserved in pure form and used for the bull testing. Farmers should be made conscious and aware about the importance of the particular breed for milch or draft or any other available unique traits, and if needed, incentives should be provided to them for maintaining a breed in puring form. And animals true to the breed should be registered for the breed society that can also act as a conservation. For ex situ conservation, we need to preserve frozen semen, at least 20 prone bulls of a cattle and buffalo, and 2,000 dosage of each bull. In case of sheep and goat, 25,000 males, 1,000 doses. These doses should be preferably stored in 2-3 different locations and devices to safeguard against any eventuality. Efforts should also be made to collect the preserve the embryo ova somatic cell at National Animal Gene Bank, and the stored genetic material of the breed can be utilized for the revival of the breed as and when required. These are the breeds which I have taken in cattle: Thar Parkan, Nagori, Rathi, Kangayam, Pawar, Panganur, Krishna Valley, Kharigad, Ongol. We are having almost 37 breed semen at NBGR Gene Bank. But if we look at the criteria that 20 bull, 2,000 doses, we are having only 7 or 8 breeds. Then what are the actions to be taken? First, we need to stop the indiscriminate breeding that is generating mass, and we are finding difficult to identify the purebreds of the animals. Then management of reproduction for shorten the service period, calving interval, and age at first calving. Then productivity can be enhanced. We should give the incentive for the elite germplant to the farmers, and we also manage the adequate feed and fodder by utilizing community lands, wasteland, or crop residue. Development of high-yielding forage varieties and popularization of food crops variety having high straw yield and development of forage bank. Regular vaccination, creation of disease-free zones and stamping out of major disease. Organizing periodic livestock health campus and awareness about our breed. And most important is the pricing for milk and milk products and value addition. We can summarize our talk that cattle are the major species for milk production in northern, northwestern, and southern parts of the country. Most of the Indian cattle breeds are reared for draft purpose and evaluated for milk production. Every time we are saying that our cattle populations are the low productivity. But the problem is that if we look, when we are looking the milk productivity, we are pulling all the breeds, whether it's a draft breed or dual purpose breed or milch breed. If we look the productivity of the only milch breed, that would be better criteria. We have registered around 50 breeds of cattle, and there is urgent need to characterize, evaluate, and register the new available population. Like in Maharashtra, if we have some new population of the cattle, we need to characterize them, and if found suitable, then register them as a distinct breed. Actually, breed is a primary unit for any genetic improvement program. Your state or central government will plan only genetic improvement program when this is a registered breed. Then we need accurate livestock census on top priority, and suitable strategy for development and conservation of Indian cattle need to be developed especially for breeds under watch. Jo breeds those are needed conservation, we need to develop conservation strategy for them. And all kind of cattle we need to conserve them. Thank you very much. In case of any query, question, I will try to respond.
Thank you, sir. Thank you very much. Energetic work with smooth, tranquil, and composed presentation. Thank you, sir. Thank you for being with us, sharing your valuable knowledge of so many years. Now I like to call upon Dr. Archana Verma, Principal Scientist, Animal Genetics and Breeding Division, ICAR NDRI Karnal. Madam's current research area is molecular genetics and genomics. Madam has worked on many research she has got many research accomplishment. Genetic variation in a number of candidate gene related to milk production and composition, suppressor of cytokinin signaling to CRH, corticotrophin releasing hormone, then mastitis toll-like receptor 4 gene, EDN3, genome-wide SNPs and SSR have been identified in Gir and Thar-Parker cattle. Genome-wide association study with milk production in Murrah buffalo is in progress. Madam involved in 5 research projects. Out of 5, 3 as a PI, and she has also worked as a co-PI in 2 externally funded projects. And she is involved in inspiring younger generation through teaching. She has guided 19 MSc, MBSc students and 3 at presently working, 14 PhD students. Transfer of technology, detection of buffalo milk in cow milk using Hansa test serum. This technology is transferred to Mother Dairy, New Delhi. A total of 123 gene bank accession number have been received for a number of gene in Sahiwal cattle and Murrah buffalo. A kind request to ma'am to share your presentation, knowledgeable presentation.
Thank you, Dr. Sir.
Thank you.
My slides are visible.
Not yet, ma'am.
अब
नहीं मैम, अभी नहीं है शेयर.
I have clicked my slide.
No ma'am, just back to the Zoom and click on the share screen button. Is it visible in the center?
Now?
Not yet.
Okay.
Are you able to see the share screen button? Blue share screen button?
No, not yet. Screen, no, it is not there. ठीक है, आ गया.
Yes. Click on the share screen button. Open your browse your slide. Fine.
Is it okay now?
Yeah, it's opening it now.
Okay.
Yeah. Can you please make it a presentation?
It's okay. It's visible.
Yeah, it is visible. Please go ahead, ma'am. Thank you.
Thank you so much for Dr. Dheeraj for your kind words of introduction. Thanks a lot for giving me this opportunity to share my views on the topic conservation and improvement of indigenous cattle. I welcome you all. And, very good morning once again to Honorable Vice Chancellor, all head of the divisions, all panelists, and all the participants. So my work has been made very easy by my previous speakers because many of the things are there which I had to say, and they have already discussed that. So thanks to all, Dr. Pundi, Dr. Jawar. So livestock sector trends in India. In India, livestock sector is an integral part of rural population. We know all that. Livestock sector alone contributed 5.21 to total GDP and 28.36 to agriculture and allied sectors. So last livestock census recorded 1.34% increase in cattle population. Means it was in 2012, it was 190.90. And now it is 193.47 in 2019. So cattle population crossbred 21%, purebred indigenous 9%, and graded indigenous 11%, nondescript 59%. We know in our genetic resources, nondescript, though 50 cattle breeds have already been registered by NBAGR, but large chunk of cattle are still nondescript or in the process of registration. So out of this, these are the distribution of means 75% non-descript. When I'm talking about the cattle, Kankrej 2%, Sahiwal 3%, Gir 4%, Haryana 4%, and others are 12%. If I talk about the milk production, so milk production, obviously we know since 1997 we are #1 in the world, but again, every time when we talk about the milk production, we feel elated. We are happy that we are #1 in milk production. But at the same time, there is something to churn about. That is, our production is highest, but productivity is less. Always that point comes. But animal growth is also there in milk production. But number of animals are even if they are less, milk production is high, but the productivity as such per animal, if we consider, we are very far behind all other countries which are leading in milk production. Means they are not as par with us, but they have less number of cattle or less number of animal heads, but more milk production. So species-wise milk production means contribution in India is like it's buffalo because it's already there. I'm focusing only on the indigenous, but in this I got this one. Cow indigenous 10%, cow non-descript 10%, cow crossbred 26%. So we always talk about that we should nowadays shift is towards indigenous cattle. We know all of us know that, but we know that is also very true that milk production, and today we are #1 in milk production, that is because of crossbred cattle. So when we talk about the breeding programs or the programs in India and overview, because conservation part already Dr. Pundir has covered, I'll just hurriedly give you an overview. That Central Herd Registration Scheme, it started in 1949, key village scheme, intensive means ICDP, and setting up of central and state government farms to raise pedigree breeding males for distribution, progeny testing, AICRP on cattle, on buffalo, network project on buffalo improvement. Then National Project on Cattle and Buffalo Breeding, is NPCBB we call that time, initiated in 2000 for integration of states' breeding policies with breeding programs for better implementation of breeding policy. So National Dairy Plan Phase 1, it was initiated under NDDB. These programs aimed at production of sizable number of genetically superior bulls, freezing of requisite number of semen doses, supply of semen at doorstep of farmers, supply of quality semen, I should say, promotion of conservation and genetic improvement of indigenous cattle and buffalo. So 10 pedigree that selection program was done, and in 10 breeds, Rathi, Kankrej, Gir, Haryana, Sahiwal, Tharparkar, these were the states where these programs were run. So NPCBB reoriented to form national program for bovine breeding and dairy development. And this had two components. One was for the bovine breeding and another one was for the dairy development. Now, Rashtriya Gokul Mission, as we all know, an initiative under this program launched to conserve and develop indigenous bovine breeds. And National Kamdhenu Breeding Center, one each in Andhra and Madhya Pradesh, not only the repository of indigenous germplasm, but also the source of certified germplasm in form of bulls, semen, and embryos. Now, we have a shift. So many reasons are there that of late we are shifting towards the indigenous breed, but there are certain qualities which they deserve. Indigenous breeds deserve to be conserved, characterized, conserved, and used in the breeding programs. They are evolved in this type of environment, thus adapted. Their adaptation is more, thermotolerance is also more. They are able to survive on low quality roughage. They are resistant to tropical diseases. The input-output benefit ratio goes in favor of indigenous breeds rather than crossbreds, as most of them survive and produce under zero input conditions. And moreover, when we talk about the milk quality, if you forget about A1, A2 also, but still milk quality is better. We know that milk fat is more, protein is more. So milk quality wise also indigenous breeds are good. But there are some reasons why these were neglected to some extent, I can say. Shift in the priority of farming community from bullock production to milch animals because of intensive agriculture and auto mechanization. Fast changing socio-economic levels of consumers, urbanization and needs of farmers, changing agricultural scenario, intensive and more mechanized agriculture, replacing draft animal power and ecological profile, shrinking grazing areas, overemphasis on crossbreeding and neglect of indigenous cattle breeds, and buffaloes also making inroads into the traditional area of certain cattle breeds. And some of the states, they are more favorable. They favor buffalo milk as compared to cow milk. This call for an immediate action for systematic conservation, genetic improvement, and sustainable utilization of indigenous cattle breeds. So what are the priority areas if we consider? It is important to produce sufficient quantities of milk to meet the demands of growing human population. Simultaneously, it is also important to conserve and improve indigenous breeds for future use. Now, developing a breeding program. Actually, a breeding program, one KISS principle, if I say, it was Philipson et al. 2011, they said, keep it simple to be sustainable. So if we talk or if we translate this in terms of indigenous, this fits properly. Long-term and simple strategies to efficiently exploit the potential to increase productivity per animal or area of land. 8 major steps, activities, choice of breeding goal, choice of selection criteria, design of the breeding scheme, recording of the animals, genetic evaluation of the animals, selection and breeding, progress monitoring, and dissemination of genetic improvement or dissemination of good germplasm, we can say, for genetic improvement. Sorry. So we can say in a breeding program, actually breeding strategy is like we can say breeding strategy tactics are designed to integrate new technologies to improve old one for the purpose of maximizing performance of existing stock. So according to that only, we have to develop our breeding strategies and breeding programs. So this very well explains this, the definition of production system, then defining the breeding goals, collection of information, because not only genomics, phenomics, as we are talking about, we know the phenotype, but genomics and phenomics, those both are to be amalgamation has to be there. Then determining selection criteria, genetic model, breeding value estimations, selection and mating, predicting selection response, consequences of mating decisions, dissemination, structure of breeding program, crossbreeding, wherever it is, we'll be talking in next slides, evaluation and genetic improvement, and maintaining the genetic diversity also, because we don't want that there should be any inbreeding also. So accordingly, we have to maintain genetic diversity, and our indigenous breeds, cattle breeds or any breeds that for that matter, they are very rich in genetic diversity. So annual genetic gain also accordingly we can get it. Now, when we talk about indigenous genetic resources, there are, means we have to prioritize the breeds, means high producer or dual purpose like that, and take efforts to cover most of the animals, then proper recording, because stress is always given on the recording. Sorry. Mega project for testing 40 to 50 bulls, use of top ranked semen in elite animals, and at least 1% genetic gain should be there. And if we talk about non-descript genetic resources, for that generally grading up, because we don't want our indigenous animals to get admixed with the foreign, means exotic breeds. So we can grade up these non-descript animals to maintain purity of our indigenous, not of the breed, but indigenous animals, grading up with the high producing or good animals like Sahiwal, Tharparkar, Red, Sindhi, Gir, and Kankres. But non-descript animals, we don't want, if we don't want crossbreeding, so our indigenous breeds are purebred, they are bred pure, but non-descript animals, those can be crossed. And use of jersey can be done, HF as we have been doing, and this is for Murrah. So this shows about the how milch breeds with reasonable good population, what can be the strategy for Gir, Sahiwal, Kankrej and Murrah. So this way we can have some like genetic gain up to 1 to 1.5% annual, and semen doses 5,000 doses per bull, as Dr. Pundir also told about this. Conservation, I'm skipping this because already he has done it. I'm skipping this one. So coming to the breeding strategies for bovine, which have been recommended by Government of India, like indigenous dairy breeds of cattle and buffalo, selective breeding. Which purpose? Milk production. Same for cattle and buffalo, drought breeds also selective breeding. Indigenous dual purpose selective breeding. But nondescript, as I have already discussed, grading up, and nondescript buffaloes also grading up. And mainly our focus is on the milk production. But of late, our focus has changed to some other traits also, which I'll be talking, like about quality also. From fat, it has SNF, protein, lactose, and nowadays people are talking about the beta carotene and all this type of quality also. So National Program for Bovine Breeding and Dairy Development, these have 3 components like bovine breeding, dairy development, and Rashtriya Gokul Mission, as I have talked. So this National Program for Bovine Breeding is to arrange quality artificial insemination services at farmers' doorstep to bring all breedable females under organized breeding through AI or natural service using germplasm of high genetic merit, to conserve, develop, and proliferate selected indigenous bovine breeds of high socioeconomic importance, to provide quality breeding inputs in breeding tracts of important indigenous breeds so as to prevent the breeds from deterioration and extinction. And second one is National Program for Dairy Development, to create and strengthen infrastructure for production of quality milk, including cold chain infrastructure linking the farmer to the consumer. To create and strengthen infrastructure for procurement, processing and marketing of milk and milk products. This is the part, means marketing, which is lacking, and that is the reason sometimes our farmer, they do not get what they actually deserve. To create training infrastructure for training of dairy farmers, to strengthen dairy cooperative societies, producers, companies at village level, to increase milk production by providing technical input service like cattle feed and mineral mixture, etc. To assist in rehabilitation of potentially viable milk federation or unions. Then Rashtriya Gokul Mission, which is too much talk nowadays, to undertake breed improvement program for indigenous cattle breeds so as to improve the genetic makeup and increase the stock. To enhance milk production and productivity of indigenous bovines, to upgrade nondescript cattle using elite indigenous breeds like Gir, Sahiwal, Rathi, Devni, Tharparkar, Red Sindhi, etc. To distribute disease-free, high genetic merit bulls of indigenous breeds for natural service. So these are some of other points, like main, I'll just talk like field performance recording in the breeding tract. This is the thing which is really missing in our system, and proper recording, performance recording has to be emphasized. Assistant to institutes which are repositories for best germplasm, implementation of pedigree selection program for indigenous breeds with large population, Gopalan Sangh, distribution of disease-free high genetic merit bull for natural service, or even for AI also, incentive to farmers maintaining elite animals of indigenous breeds, which Gopal Ratna and all this type of awards have already been instituted. Organization of milk yield competitions for indigenous breeds, just to encourage them. They should get some incentive so that they will go for the breeding and maintaining indigenous germplasm. So for pure breeding, Red Sindhi, Sahiwal, Tharparkar, and Gir. These are efficient milk producers, so obviously these should be purebred. There should not be any admixing of any other breeds, so that we can have pure, pure animals, true to the breed, which we call as true to the breed. Then selective breeding, expect genetic gain 1 to 1.5% per annum at organized farm, and in farmers herd it is comparatively more. Such breed improvement programs must form coordinating bodies both at organized herds, including progressive goshalas, as well as farmers herds under field conditions. Sorry. So elite female should be maintained for production of future bulls, which we can say that bull mother farms can be maintained. Young male calves selected on the basis of their dams yield and physical appearance. Progeny testing of these bulls under field conditions. Female and male calves should then be selected out of these bulls and brought to the elite herd to repeat the cycle. That way we can have bull mother farms and good male germplasm we can prepare. So for grading up also, majority of the indigenous cattle survive in areas where feed and fodder resources are negligible, climatic conditions are extreme, and agriculture production is very low. So nondescript cattle should be graded with the superior breeds to improve their productivity. Choice of the improved breed, like according to the specific requirement of the particular region. Why I'm saying this, or why it is quoted like this, is when animal is bred in their own tract, home tract, obviously, then they perform better. For them, adaptation is better for them. So, and state and duty-wise breeding policy of the country, like Andhra Pradesh, they have established breeds, and for established breeds, grading up. Arunachal Pradesh and Assam, grading up, and cross-breed with Jersey. Bihar, there is some sound. Bihar and Jharkhand, local cattle, grading up with Tharparkar, Haryana and Red Sindhi, and crossbreeding with Jersey. Chhattisgarh also, same. Like these are the, I have taken this from our DHD site. Gujarat, Gir, Kankris, selective breeding, and nondescript, obviously, grading up or crossbreeding. For Goa, local cattle, grading up with Red Sindhi and crossbreeding with Jersey. Haryana and Sahiwal, selective breeding they are doing. For nondescript, if there are some nondescript breeds, nondescript animals, they are graded up with Haryana, Sahiwal and Tharparkar. Maharashtra, Khilari, Dangi, Golo, Nimari, all these selective breeding and nondescript, either grading up or crossbreeding. Himachal and Jammu Kashmir, local cattle, grading up with Haryana, Red Sindhi and crossbreeding with Jersey. And HF, even Punjab, they are keeping HF, pure HF also. So similarly, Karnataka, they have got very good Dharani, Krishna Valley, Khilari, Amrit Mahar, Halikar, this for selective breeding, and for nondescript, grading up and crossbreeding. Similarly for Kerala also. Now, there are some steps which we have to take for, as we know, as I have discussed, that recording system or genetic evaluation system has to be there. So National Genetic NGES, as we say that, why it is needed? Provide uniform genetic comparison to maximize accuracy of prediction of genetic values and animal rankings, and an essential selection and mating tool for unibreed and multibreed populations, a marketing tool that increases the economic value of the evaluated animals. There are some molecular genetic techniques, or some techniques, I should say, like traditionally we are selecting animals using phenotype and exploitation of genotype through phenotype. But current techniques directly based on genotype. Now, future technique is genomic selection. So marker data combines with phenotypic and pedigree data to increase the accuracy, a reduction in genotyping cost and GI, increase the selection gains per unit of time, and lead to double rate of genetic gain. So very happily, like very recently, this IndiGow1 chip has been developed, indigenous chip has been developed for in the process of this Atmanirbhar Bharat, we can say, and that will enhance or that will add to the doubling the farmers' income also. Developed by, and it has got 11,496 markers. So we can select animal based on all this type of markers. 43 breeds have been used in this, and totally it is indigenous. So something it is very to be proud of, that it is a totally indigenous chip for the indigenous cattle. So what is this? Is purely indigenous, as I told. Chip is to achieve goal of conservation of our own breeds with better characters and help towards doubling the farmers' income by 2022, as our government wants this. The manufacturing of this chip is in synergy with Rashtriya Gokul Mission and a great example of Atmanirbhar Bharat. Further, the chip exemplifies the application of scientific knowledge and innovation. But we know that it is not so easy. That much amount of money we don't have, that budgetary requirements are more. Being costly affair, we have to go long way to implement and achieve success in genomic selection strategy for livestock improvement using this chip or the genomic selection things. Till then, identification of significant SNPs for milk production, mastitis, reproduction, thermotolerance, and of late linear type traits or other conformation traits also. We have done at NDRI. For milk production, almost I think around 12 genes, mastitis 32 genes we have done, reproduction 11 genes, and thermotolerance also 9 to 10 genes, and linear type traits we have done around 19 genes. So if we club all that, the significant SNPs have been identified. If we club all that information, so we can develop a test, simple test also. By the time we go for the chip and genomic selection type of thing, we can identify, we can go for the RFLP test or this type of some test, which will identify our good germplasm for particular trait. So these are some now there are some constraints also. I will not go very much in detail because it will take a long time. I think. So there are some constraints which because of low, we don't have many things which are required to take up this type of work. These are the constraints. We don't have fodder, we don't have bulls particularly, because we require many semen doses if we go for artificial insemination. Right now, only 33% of our population is covered under AI, but in next 5 to 10 years, we say that it should be 50%, at least 50%. So that, why I'm saying that, because in that, if to farmers, if good quality semen is not available, then they will simply go for natural service, and whatsoever animal is available or bull is available, they will get their cows inseminated. Obviously, then their genetic improvement is hampered, and moreover, production is also compromised. So new technologies which can be, along with all these breeding strategies, some new technologies have to be in, we have to incorporate, or we, sorry. I want to go back, sorry. This one. Like, sorry for the interruption. Newer breeding technologies like marker-assisted selection, genome-wide association studies for faster implementation of various breed improvement programs and for increasing production. New technology of like IVF, as Dr. Subhash also told in the morning, that those technologies, assisted reproductive technologies, can be clubbed with all this, and semen sexing technologies to provide greater choice regarding breeding to the farmers. Actually, why semen sexing technology is so important from my point of view, because whenever there are some discussions, we say stray male animals are there on the roads and they don't have any future. Nobody, even Goshalas will also not accept those animals. So it's better we go for semen sexing technology so that we have all the female animals or those males, only those males, we allow all those males which have good genetic merit, so that we don't have stray males to die on the roads, which is really very touchy at times when we see this type of animals there. So the delivery of breeding services would be regulated by fixing standards with periodic evaluation of service providers. So strategies, same, like we have to clean milk production, we have to give some emphasis, hygiene, quality control, and strengthening of dairy cooperatives and diversification and value addition. Field data recording, as I have already told, like it is really need of the hour. Without that, because genomics have to be mixed with phenomics, without our recorded data, it's very difficult to, even for genomic selection or advanced technologies also, we need this type of strategy to be followed. But there are some constraints in data recording. Farmers reluctant to reveal the production of their animals. Sometimes identification is expensive, increase in recording cost with the distance between herds, loss in data from heifers due to migration, sale, and change of ownership. Because farmer will sell as soon as he feels that my animal is not producing as per his expectations, obviously he will sell that animal. And then information network for animal productivity and health, enough, which was developed by NDDB in 2008 for database of performance recording. Area under fodder and pastures, that is also decreasing. That also we have to see. Health services, if animal is having some delayed puberty and stress and repeat breeding, they should have immediately consult veterinarians. So veterinary services should be available in the nearby areas for them. Feed and fodder availability, that also I have discussed. So now this is a very matter of concern for all of us, or for breeders, because that's a very big question mark, very big question before us, that how to meet the requirement of breeding bulls. So like, according to an estimate, to cover even 30% breedable bovine population through AI, we need 1050 proven bulls of crossbred cattle, 5700 proven bulls of well-defined indigenous cattle, and we breeders, we know how long and how cumbersome it is to prove a bull. So we can understand that at what verge we are standing at this moment. It's a really difficult one. This is the total demand, means how much. So establishment of bull mother farm, as I have already discussed, of improver breeds to ensure availability of quality male germplasm. So quality of bulls, obviously we, in nutshell, I can say for quality of bulls that dams milk yield should be good. Second, animal should be evaluated properly. Like first, it has to be progeny tested, and but for that we require many daughters to be born in the farm or in the field condition, and we should track them. And tracking is not so easy. For that, maybe we can use some sensors or this type of thing, or some connectivity, some linkage between farmers and farm and farmers, so that can come in a common computer, and we can get everything, all information of their daughters, bulls' daughters, so that we can select the bulls in future. So extension of field AI network through MED3, that is also one of the things. Technical, I have already, this poor conception rate is another issue which we have to address. Reason being, there are many factors for that. So everything has to go in coordination. coordination in the sense like when animal is coming in heat, immediately means that time they should have veterinarian or any inseminator, to be very specific, at their doorstep, so that they can they will not miss that particular cycle. Missing a cycle is a very means economically it affects the farmer, which should not be there. So newer generation, this I have already discussed. Social constraints are also there. But now that thing has shifted from buffaloes or crossbred to indigenous. That is also very positive point, we can say. So there are some recommendations which we can make when we think about the breeding strategies, or in our context, in Indian scenario. So superior breeding bulls should be identified and evaluated through large-scale collaborative progeny testing programs, associated breed-specific multi-herds. Herd registration and animal performance recording system at farmers' herd should be developed and linked with PT programs, progeny testing programs. Nucleus herds, bull mother stations, and germplasm centers for different cattle breeds should be established for production and dissemination of superior germplasm. Closed nucleus herds of crossbred cattle wherever it is required, because altogether we cannot say that switched off, crossbred is switched off. It is not like that. So well-defined indigenous cattle and buffalo breeds should be adopt ONBS program with or without MOAC for faster multiplication of superior germplasm. So embryo splitting, cloning, as in the morning also it was discussed, these assisted reproductive technologies are to be used for the same purpose. So DNA marker studies for identification of genes of interest, like milk quality, quantity traits, fertility, draft, disease resistance, thermotolerance, all these things, and marker assisted selection as new breeding tool need to be explored for identifying superior animals and commercial exploitation of indigenous breed. This is, we can say, as a stopgap arrangement, what is happening now, and what we are thinking in the future. For future, we are thinking genomic selection. So that let that be make as a bridge for these two things. So breed society should be established to promote self-participation of farmers, breeders in the breed conservation and improvement programs. For draft or dual purpose cattle breeds, intensive utilization of bullocks for draught power should be promoted through better designing of agricultural implements for efficient and economic work output. We know everything, almost this plowing and all these things are mechanized to great extent, but still draught animals are required. That is not 100% coverage is not there. So better marketing of animals and value addition, milk, meat, and other animal products, which are make animal rearing more economical and commercially viable. This is the need of the hour, that whatever we ask a farmer to do, that should be economically and commercially viable. Then only farmer will adopt that. An apex body at national and state level should be established for overall monitoring and implementation of breed conservation and improvement programs. To make available the superior germplasm and breeding facilities at the doorsteps of small holders, infrastructure facilities such as AI center, animal health centers, milk procurement and extension centers, fully equipped with trained manpower should be developed. It should be provided to them. So to conclude, I can say it means we have to strengthen the herd. So increase the proportion of animal under AI from existing almost 30% to 50%, infrastructure development, monitoring, conventional selection, along with new generation technologies at organized herd, which may be like right now, even ETT or these things can be extrapolated, or that can be translated in the farmer's herd also. Means that can go from organized herd to farmer's herd. So we have to strengthen the frozen semen production infrastructure to produce quality semen to meet the requirement of increased AI coverage, and produce bulls of high genetic merit for semen production through progeny testing. Then only we can say that we are number one in production, but we are number one in productivity also. because that is always that boggles everybody, every reader's mind, that where we are going, where we are and where we want to go. So for that, we have to make the bridge and we have to travel along for that. So take home message is select the animals as per the breeding goals, genetic merits and accuracy, sustainability, which is very, very important, that whatever we say that should be sustainable. It should go for a long time. And quality and value addition, because nowadays even cow dung, cow urine and all these, particularly for the indigenous breeds, we can say that value can be added, and the Panchagavya or this nowadays this Panchamrit or these things are being used even for the natural farming also. This proposition, these compositions can be proposed to be used in the field conditions. So we have to give more and more stress to the quality, so that our farmer, ultimately we are working for farmer, and the farmer should be happy to breed animals. They should go for animal husbandry part, and they should be animal breeders, or they should maintain animals, not that they will get distracted from that. And that will only happen when they are earning money from that. So these things we should take care of. So thank you very much for the patience hearing to all of you and organizers for giving me this opportunity. And thanks a lot. Thank you very much.
Thank you, ma'am. Thank you for the insider insight, calibrated blueprint for making true masterwork. A feel of sublime serenity after your presentation. I'm a big thank you for all three speakers for sharing your views, and we are looking forward for guidance from you in the future. With permission to the chairman and the president of the session, we will proceed for the panel discussion. As a Lao Tzu, a great philosopher, said that the 1,000-mile journey begins with a single step. I request all panelists to put forth a step by sharing valuable, constructive, and sparkling views. I request all panelists to share their views, please. Dr. Zawar, sir, one question was there from Narayan Rao. Sir, I'd like to know how this technology help in genetic diversity in cattle. So we'll start from there.
I mean, for the genetic diversity, you can say, for example, you are using one good donor cow, looking at the milk production of that donor cow, which is a very good elite cow is there. And whatever oocytes which are being collected, even those oocytes, they can be fertilized. If you get a good number of oocytes, you can use different bulls, you know, and using the semen of the different bulls, you can have the one single donor and the progeny from this way, one of the example of you can have a genetic diversity from the same animal can be done.
Speaker
Shall I ask the question?
Yes, sir, you can proceed, sir.
My question is to both the speakers, particularly Dr. Pundir sir and Madam Verma. First of all, I am very much thankful you have accepted our invitation. So very much thankful to both of you. Sir, my question is that, and it is one question many times asked by the plant breeders, that shall we go for the crossbreeding within the indigenous breeds? That means, can we cross the Gir with Sahiwal and vice versa, or other breeds also? So, shall we go for it, or shall we avoid only for you? We go for the selective breeding as per our breeding policy.
Can I reply?
Yeah.
Yes.
In my view, the purebreds should be improved by the selective breeding, pure breeding, not intercross. And while in case of nondescript, a particular breed of that region that is useful, you can upgrade the nondescript, but well-established breed should not be crossed.
Thank you, sir.
No, sir, our plant breeders, as they are saying that everybody, when we goes to the research council meeting, at that time, one plant breeder insists this, why we should not make crossing between our indigenous breed, and it is not for our policy point of view, but for gain. In case of our, just like in germplasm, we are getting some good types of germplasm for yield and quality parameters. At that time, those good parameters available in suppose in Gir, Saiwal, and Thara Parkers, we can. Why we should not go to this crossing program in this animal component?
In my view, the concept of breeding in plants and animal is entirely different. When you are taking the production repeatedly, then this is not desirable. When you are taking production single time, then it can be. Like in meat animal, if you want to pick, for F1 you are going to you are not going to take the production from the F1 repeatedly. But in case of cattle, you need to take the production repeatedly from the F1, F2, F3. So if it is for the meat purpose, you can go, but for repeated production like milk production in different generation, in different F1, F2, F3, it is not desirable.
Sir, one more question. Sir, what are the possibilities of getting the heterosis when we cross the Gir with Saiwal?
Again, the same reply. When you are taking the production repeatedly, it is not desirable. And heterosis for the other traits, for F1 meat production, and you can go for heterosis.
Yes. So only meat production traits you can go for this, or
in
for
different no, in different generation, you have to get the loss of the productivity.
So one question from me, sir. I'm Dr. Shivkumar Patil from Ravuri Sim Station, sir. My question is to Dr. Jawar, sir. This question is related to GST, sir, actually. As you said, sir, ET and IVF is very wonderful for genetic gain and breed improvement. But sir, as you know, sir, actually some GST or hormones, equipments, consumables, which is imported from foreign countries, actually we paid some custom duty on same one. Even at the time of sale of embryos also, it attracts some 5% GST on embryo sale and service, some 18% something GST, sir. Is any help we can get from government to sir, waive off this GST on this thing, sir, so that this technology will be cheaper, sir, and reach to the maximum farmers' door, sir?
We are actually presently we are working on that, and this is one of our agenda points, and we are discussing with the Secretary Animal Husbandry, Government of India, the CMU which has been formed. This is one of the agenda we are working on it, and I hope some solution will come out of it.
Thank you, sir. Thank you, sir.
Hello. I want to ask, hello.
And Dr. Aswal, go ahead.
Yeah. I want to ask one question to Dr. Verma, madam. That the genomic chip, is it now available? Means if we can send our samples, so is it in the now working phase? And what is the reliability of the chip, and how many SNPs you have included, madam? I'm working in ULDB.
That is done by Hyderabad, NIAB, National Institute of Animal Biotechnology. And they have 11,496 markers they have on that. So very soon it will be available. I don't think it is marketed right now, or it's available, but we can just talk to them and seek the possibility.
Okay, so NBGR is also they were also developing chip. Is that this Indo-GAU chip is the same one, or that is different one?
No, it is different. Actually, in India, 4 agencies have developed the chip. One is the NBGR, one is the NDDB, one is the BAF, and one is the Hyderabad firm. So it is in process, it is going on that all the chips, they merge the data and develop only single one. It is going on in process. But for NBAGR chip, it is not available. I think NDDB chip, I think it have 50,000 markers that is available in market. But for NBAGR and NIAB, I think they are not available now.
NDDB, it is available.
NDDB it is available.
Indigenous, IndiChip, that is known as IndiChip.
They have very few markers. Not
Sir, Dr. Pundi, NDDB chip is also not available. That is available with them.
But many time in discussion they are saying it is available.
No, it is not available. You check that. I've already checked, but it is available with them. That is not available in the market.
Hello.
Yes, sir.
My question to Dr. Shyam Javer, role of IVF. Sir,
please go ahead.
As you have mentioned the wonderful technology, IVF and all, there are some constraints. Just like every time we are facing the problem of the good donor for the selection of gametes and all. So you are a mentor, Government of India, any kind of the financial assistance for genomic selections so that the good donors can be available with certain defined traits?
You have a very good point, sir, about the donors. This was really a very critical issue about the selection of the donors, and different options have been worked out. You know, there are farmers who may have the good donors, but they may not sell the donors. You may not get those donors. There are some excellent goshalas are there where they have a very elite animal. So what has been done, I mean, some people, they offer the different ways we can take the oocytes from these donors and pay them for one extraction. You know, if they are confident of their team, that you pay them about 5,000 rupees for that 15 minutes OPU, where you collect the oocytes, and the farmer has to be pretty sure that there is no damage, which normally doesn't occur with that cow. Second one, another choice is that wherever the farmer don't want to sell the good donor cows, you can share the embryos. Whatever embryos which are produced, they can be shared 50-50% embryos amongst them. Third one, you know, in case of MOET, you can't use the animal with the reproductive problems, infertile donors. But in case of IVF, there is no problem. Even if we have worked on some of the donors which they were not getting impregnated for almost 2 years, even with AI, natural breeding, everything. But they can prove to be a good donor as a IVF donors, because in case of IVF, you are just utilizing the ovary and you are not touching any part of the reproductive tract. So this way you can get the good donors from the farmers. Another choice is that leasing out. You know, you can request the farmer to lease out his donor for about a year or so, and in return, when you return the donor back to him, you impregnate that donor, and along with that, give him about 3 to 4 female calves along with it. So that's another way, and you can extract as many oocytes you want over the year and produce the embryos out of it. So these are the different options we have been thinking, and it has been implemented also. Thank you.
Okay, thank you, sir.
Hello, I have one question to Madam Sharma.
Yes, sir.
Madam, you mentioned that indigenous breeds have very rich genetic diversity. So can we go for this pedigree selection among with these indigenous breeds for a specific trait?
Is it to me, this question?
Yes, sir. You can continue, sir.
Okay. In case of pedigree selection, the response or the gain is very less. That's why we are not going. We are suggesting the position testing or genomic selection. You can go, but gain is very less, pedigree selection.
Yeah, it is due to the by rules or by policy. we are denying the crossing between the indigenous cow, or it is a result. The result of the milk yield will be reduced or not affect. Is it any experience on that matter in this aspect you have?
We got the result from the crossbreeding. In crossbreeding, what happened? In next generation, after F1, there is a loss of productivity, reproduction failure. They are not conserving. So we need to learn from the crossbreeding experiments.
These will be problems. Surprises. Okay. Our second thing is the in market, protein is very important because in crossbreed cow, we are observing the 34% proteins per percentage, 34 to about 34.5%. In indigenous cow, it is increased. It is a high proteins we are observing. And in buffalo, more than 36% proteins in milk industry has getting this good benefit from buffalo milk. And that's why how to increase the protein percentage of the milk of the indigenous, or I think that crossbreed cow is giving the very high yielded, and we want to transfer this the gene, that is the high protein gene, from the local cow to the crossbreed cow. How it is possible?
I think there are technology. They are not available in our country. Gene transfer like this technology is there, but they are not available in our country.
It is not available.
Country, it is not available.
Yeah.
It is not simply crossing. It is another technology.
But how to increase the protein percentage in our indigenous milk, as like as buffalo?
The only way is the selective breeding. You select the animals, those have the high rich in protein, and breed them, select them.
If you allow, I can have a comment on this.
Yeah, because
Yes,
the
sir.
price of the milk product is depend upon the protein percentage, and that's why we want to enhance the protein percentage. Please, sir, you can give the other sir. Please,
Yeah,
you can
thank
add.
you very much. So I'm Dr. Sunil Jadhav. I'm Principal Scientist, Animal Nutrition at Indian Veterinary Research Institute. So thanks to the Honorable Vice Chancellor and the organizing team for giving me the chance as a panelist here. So the particular question of Dr. Gadak sir, that how we can manipulate the protein content of the milk. So though the genetic composition of animal have a bearing on this, factor which is having a direct effect on the milk percentage of the milk. So rather than I think going for the transgenic, what the way ahead Dr. Gadak was talking about, I think it's the nutritional manipulation and nutritional formulations have a strong bearing on the protein. So it's all about the protein content of the diet mostly. And then of course the digestibility capacity of the animal is another issue. So I think what the Dr. Verma ma'am was talking about, the genomics. So if we correlate the nutrigenetics, that how the genetic profile of animal have a bearing on nutrient utilization, I think this should also be one of the component. In the one slide she was talking about the KISS, that approach, it should be simple and sustainable. And under that some 8 points she was just narrating. So if we again consider while selection of the animal, that how the capacity for digestion of the nutrient, and this can be one of the parameter for selection of the elite animals or selective breeding, or even for that reason when we are going for upgradation. So though this today's talk is on the genetic improvement and lot of the concern and discussion on the genetics, but as a nutritionist and as a member in the panelist, I also take this opportunity, if I'm permitted, to highlight few points. So as we are talking about the indigenous cows, they have very unique capabilities. They are disease resistant, they are thermotolerant, and we also discussed that they have the capacity to utilize the roughage efficiently than the crossbreeds. So though it is true, and today we are discussing about their conservation and selective breeding, But you will agree with me that as and when we increase their production potential, it is going to hamper their reproductive efficiency. So both are inversely proportional. So if you are planning to increase their performance, genetic constitution, we should have the manipulations with regard to their nutrition. We should have proper strategies. That is one of the researchable area. So this center, I hope, my understanding is that it's going to have a better research outcomes in time to go. So if we at all we are improving the genetic potential of animals, we need to have to develop the proper nutritional requirements, because the present ICAR nutritional standards, they are mainly based on the performance of the crossbred animals. Those are medium to high yielders, and probably our indigenous stock in time to come, we may not expect very high yielding milk production. So accordingly, we have to match our nutrient requirements. That is one thing. And we have listened 3 learned speakers, as Dr. Shyam Dhawan was also talking about that the indigenous cows are prolific oocyte producers. So my concern here is that their reproductive efficiency may be certainly very good and very high. But as we know that nutrients, they have profound effect on the reproductive ability of animals. Dr. Pundir also told that we need to shorten their calving interval, their age at first calving and all. And among this, I think nutrition is going to have a very important influence on these parameters. Mainly the minerals, vitamins, the need for cell replication, cell division, so oocyte performance, ability of spermatozoa to further carry out fertilization and all. So I think under researchable issues, as far as nutrition is concerned, we need to customize the nutrient requirements under the specific environmental conditions, because these animals were also taken from one breeding tract to another breeding tract. Their adaptability at that particular stations in that particular environment. So I think as a nutrition point of view, in this today's discussion, I may be one man odd in this discussion, but as far as the nutrition is concerned, I think these are some of the points. And also if we see the indigenous cattle, they are more efficient of fiber utilization. And it is also discussed that their methane production, which is one of the important greenhouse gas, they may be having the sustainable effect, or we can say that their methane production may be higher or lower, but in that case, it is now being worked out that the human microbial profile of indigenous cows, it must be having the beneficial effect as far as the greenhouse gas emission is concerned. So in that way also, if we are going for conserving, improving the desi cows, indigenous cows, all these points should be taken into consideration as far as the nutritional strategies or nutritional research is concerned. So with these few points, I thanks for giving me the opportunity as a panelist, and as far as nutrition is concerned, if some queries are there, I would like to sort it out or discuss with help of all the learned participants in this.
I'm Dr. Ulmek. I would like to respond to Dr. Gadak's question.
Yes, sir, please.
Well, when you talk about the crops and the animals, the biggest difference is that the generation interval in crop and the generation interval in the livestock is different. So thereby you can have more number of varieties in a short period of time. But for the animals, it takes generations together. It means if you see the evolution of the different breeds of the livestock, it may be all indigenous cattle. It is the history, especially when I've studied for the Khillar, I know that the history is about 500 years old. The Khillar breed has been evolved over a period of 500 years. So whatever the characteristics which have been fixed in that breed over a period of time, based on the climate change, based on the available natural resources as a feed and fodder, and the priorities of the farmers. So accordingly, the breeds have been evolved. So as you are asking a question about crossing the indigenous breed, that is not going to help us. It is only Bos indicus and Bos taurus where you find that the milk production in the indigenous cattle has been improved. Now, what is important is that whatever the non-district population, which is a major population in our country, so that needs to be upgraded with our indigenous breed, and it is rightly said by Dr. Archana Sharma, Archana ji. So this is the only thing, and I think that we should not confuse like crop sciences. In animals also we can do the same kind of improvement. That's not possible.
No, sir. One my doubt in my mind that because we are drop purpose animal, then milk purpose animal, and dual purpose animal. We are classified this animal. But only I am thinking about the only milk purpose animal. Those indigenous breeds are very good in milk production. And we just like this is my I may be wrong, but my thinking is the only for milk purpose we can cross these breeds only milk animals in crossing program, and we can we improve the milk production or otherwise milk quality also. That is, we can say that 14%, because 1% protein increases the value of the milk product is about 20 to 25 rupees per kg. And that's why my intention is that.
But basically, we have to understand.
So many days, I am thinking on that. I'm asking this question to my scientists also. But this is a proper forum because you peoples are here, and we can clarify this. My doubt in my mind.
Yeah, it is a generation interval is a basic fundamental thing where you cannot compare animals with the crops. In the crops, you can have several varieties within a short period. But for the animals, I said that it takes hundreds of years to evolve that breed. Because we have already studied at Mahatma Phule Agriculture University, we have already studied the different indigenous breeds, like it may be Khilar, Pandharpuri buffalo, Sangamneri goat. And I think that our university is topping so far as the developing the breed descriptors for the different indigenous breed. Now, and as a result of it, now we have the network project on the Pandharpuri buffalo. So thereby we are spreading the genetic material to the field, and whatever the field testing which has been done, I think that it has got very good performance. That is improvement in genetic improvement in the buffaloes.
This, as per your suggestion, there will be no improvement in the genetic gain will not give then.
No. You see, we have to upgrade the non-indigenous, non-descript animals.
Okay, this
indigenous breeds of the cattle. That is the only solution.
This is, I think, in the plant breeding, this is selection procedure. Selection procedure, we are following this.
Yes. And you see, the generation interval is also very small in the crops.
Selection procedure, we have limitations.
Yeah. That limitation is there.
In this breeding program, we will get the genetic gain. That's why we are assuming that it will be possible.
Right. So upgrading is the only way for that is non-descript population. That is the biggest population in the country, which needs to upgrade it with the indigenous breed, discrete breeds.
Yeah.
This is one thing.
We want to conserve our breed. That is no problem. This is
no. That has to be a priority. Now you see that
selection method we are going forward. But in the old days, some suppose there is a daughters delivered at that time from one area, one cow, lacting cow, the cow is giving milk. They are giving to this daughter, and they are supplying this one village to other village, one place to other village. And at that time, these breeds are transferred from anywhere in our country.
I just want to add one thing, with the indigenous breeds. Definitely, if it is done with a proper selection, you can see what Brazilians have done by taking our gear and how with the selective breeding, how they have reached to the very good milk production records from the gear itself, which is our breed. And a time has come now for us to import the semen, embryos, Red Sindhi, they have developed it very well. So our own indigenous breeds, we need to preserve it, as rightly said by Dr. Umang, that we should preserve it rather than going for a crossbreeding with them. We have got a lot of scope to use the crossbreeding, upgrading with the non-descript breeds. We have got such a high percentage of them. Thank you.
Okay, thank you, sir.
Second point I'd like to highlight is that in the change of the climate change situation, we need to develop a sustainable policy. It is not simply and for the sake of the technology, we have to introduce that. It needs to sustain it. And when we can sustain it, if we are using our indigenous breed, whatever the and the conservation process is not also proper. We have to develop the nucleus herds we have to develop, breeding farms we have to have it, And this has to be discussed with maybe at a government level. And unless and until we come out with a policy matter, we cannot bring this specific and definite genetic improvement in this population of the livestock.
Sir, one more question to Dr. Pundir, sir. Hello.
Yes.
Yeah, one more question to Dr. Pundir, sir, R.K. Pundir, sir.
Yes, please.
Sir, in general, how much genetic gain we expect in pedigree selection? And another question is that, say with 20 daughters, how much of genetic gain we expect in PT project instead of going to 60, 70, suppose we go for 20 daughters. So how much genetic gain we expect in terms of, as compared with the pedigree selection? And another is my point is that instead of saying non-descript, we can call it as non-described, because over the period slowly we are having 20 beads and 40 beads. Once we will have 100 beads also over the period of time. So instead of saying them non-descript, we should call them non-described. So this is my suggestion. But first two questions, kindly, sir, please, Dr. Pundir, sir.
Yes, in case of progeny testing, we are expecting 1 to 1.5% genetic gain per generation in progeny testing. In case of pedigree selection, it is very less. While in case of genomic selection, we are expecting high rate of gain because generation interval is reduced in case of the genomic selection. In progeny testing, genetic interval is not reduced, generation interval. So we are only expecting 1 to 1.5% gain in progeny testing, while in case of pedigree selection, it is very less. Well, second question is your non-described and non-descript. Actually, now the awareness has come up. And in 2008, we have only 30 breeds. After 2008, many universities, many ICAR institutes, including NBGR, they have described the population. And proportion of the described and non-described is reduced. If you look 2007 and 2013 breed census, in 2007, we are having around 69% non-described population. In 2013, we have 59% population. So now awareness has come up. And that time only 37 breeds were available. Now 50 breeds are available. So whenever the last breed census will be there, definitely further decline is there in the non-described population. And it is true. In case of non-described, we can say it is non-described because we have not described. That's why they are the non-described. Thank you.
And this will have a huge impact, sir, as because we want to cross the non-descript animals, say so-called, with the Bos Taurus semen, suppose. So what is happening over the period, slowly, some of the breed population is drastically reduced because of these definitions, I feel.
No, major problem is the reproduction. When you cross you can look the result of the crossbreeding experiments. Then you will get a better idea. Why we are saying grading up of the non-descript population with the established breeds of our country? Because of the adaptability, there are so many factors, resistance for disease, hot, and adaptive to different climatic regions. That's why we are suggesting that the non-descript population will be upgraded by the well-established breed of that particular region. So such type of problem should not come up. Thank you.
Sir, thank you. So there is one question from Dr. Prakash Kadar. How to increase milk productivity of khillar, because the low productivity of khillar farmers goes for the crossbreeding with the HF.
Either genomic selection, it is not like this is there from tomorrow you can start. You need to infrastructure, you have to develop. It is the only selective breeding. Selective breeding means in khillar, you have the range of the milk production. There may be some animals, those are producing only 1 or 2 kg, but maybe there are some animals, those are having 8 to 10 kg milk production. So from these 8 to 10 kg animals, you select the bulls immediately and go for the breeding. And simultaneously, you also develop the facility for the progeny testing. Once you develop the facility for the progeny testing, it will act as a reference population for the genomic selection. In genomic selection, what we are doing? We are genotyping the animal for large number of the markers in lakhs, 6 lakhs, 7 lakhs, like this. Then we are correlating these markers with the production traits. And based on these results, we are selecting the animals, but it will take time. Immediately, you need to identify the animals, those are good in milk, and take them as a breeding males. Immediately, you can do that.
Okay, sir. So next question, can you upgrade ND population on basis of color pattern? Because in Maharashtra, there are different color pattern. So can you cross white color cow with Tharparkar? Can we cross a brown color cow with Sahiwal or Red Sindhi? Or can we cross a mixed color cow with Rathi for ND?
Actually, if you're looking for the production, this is not desirable. But if there is a specific choice of farmer that I want only that color of the animal, then you can go. Otherwise, for production, it is not like this. But if some farmer have a specific choice that I need such color type of the cattle or cow, then I think you can go.
But sir, milk production also increase by crossing with this milk type breed with the ND.
Yes, definitely. If you cross a good breed, non-desert with a good milk producing breed, it will definitely increase. Okay. Major problem in crossbreeding of indigenous and exotic is the reproduction failure and the adaptation.
Sir, another question, how you can identify pure breed at a field level by using, is there any test for identify Sahiwal or Red Sindhi?
If you are thinking that you can identify the blood sample, it is not possible. Only the physical traits. All registered breeds have a breed descriptor. Based on the breed descriptor, you identify the trait and you match in the field. This is the only way. From the blood, you can't identify. Even we can't differentiate all 50 breeds based on the DNA or blood sample. Even today, we are not able to differentiate that. That's why in case of breed registration, only the physical traits is kept, not genetic traits, genetic studies or genetic marker studies.
Okay, sir. Another question, sir, if you see the data of EI, sir, mostly in indigenous cattle, farmers are preferring bull only. Only 10 to 12% farmers are using EI. So what is the comments on that, sir, particularly?
We need to create the awareness among the farmers. We need to tell them the advantage of the AI. AI means you have the semen strain of a known bull, non-production bull. In case of artificial insemination, you don't know, or very few know. But in case of AI, the semen strain, you know which type of animal, which bull mother is there, what production level was there. So if you make aware them farmers about the production and productivity of the semen strain, I think farmer will get.
But sir, as per the results getting to the farmer, in a crossbreed we are getting very good results, but in indigenous cattle it is not. So then what?
Crossbreeding AI?
Yes, sir.
I'm not able to understand.
Sir, in crossbreeding we are getting good results of artificial insemination, but in indigenous cattle it is not happening, sir, in somewhat at farmer level.
I think it is the question of the reproduction, and in AI process, frozen of semen and semen taking, and they are making the AI, so skills is required. So maybe some skill is there. Otherwise, I don't think it is.
Okay. And sir, last question, is there availability of PT bull semen for the farmer through NBGR?
Pardon?
Progeny tested bull semen is available for farmer.
No, it is available in CIRC Meerut.
Okay.
NDRI Karnal.
Okay.
Not at NBGR. For NBGR, we are keeping only for the conservation purpose. Okay. For commercial purpose, it is available in CIRC. CIRC also has a good cattle institute in Meerut. They are also having a good tested bull semen in NDRI also. So you can take from there. Even NDDB also have good semen.
Okay. Thank you.
Which breeds you want to know about?
Sorry, ma'am.
Which breed? Semen.
Sahiwal, Gir, Rathi, Tharparkar, Red Sindhi.
Sahiwal we have in NDRI and Tharparkar also.
Okay.
And
CIRC has a एक मिनट, CIRC Dr. Umesh, you can.
Dr. Umesh Singh. Sir, you will get the semen of this Gir as well as the Kankrej and Sahiwal. That progeny tested semen, there is no problem. We have. Another your question was about that AI. AI in indigenous at par with the crossbreed. We are already running 3 projects. Genetic improvement of Gir, Kankrej as well as the Sahiwal. There is no problem. Even in Gir and Kankrej, we have already registered farmers animals. We are already working in the field and we are getting near about 48 to 49% consumption rate. Here there is no problem in consumption rate of indigenous as well as the crossbreed. Another problem, you have already raised the question about the farmers. Why they are not interested? Problem is that we are not providing the facilities to the farmer's store. Farmers are always ready. If we can provide that facilities, AI facilities with good germplasm of any breed, now farmers are ready to accept it. But still problem is farmers, they are not getting the facilities. If that facilities will be available, farmers will move from that old system for the AI system. Another problem is non-availability of the good germplasm in that region. That should be available. If good quality of the bull semen is available, naturally farmers will accept. Another question is the non-described animals. Now our objective is to upgrade the non-described population, non-described population by use of the good quality germplasm of the defined indigenous breed. We can upgrade them, we can divert, we can move from one population to another population. Suppose in Gir pocket, Saurashtra, we are having the non-descript population. Why not we can go for the change of that population into the Gir? Continuously, we can use the semen of Gir with that non-descript population. After 4 and 5, 6 generations, we can get the whole population that will be diverted into Gir. Likewise, we have to implement that policies. Already our Dr. Pandit sir, Dr. Madam already spoken about that. Crossbreeding, that is not applicable, not permissible in our indigenous cattle. And that is not comparable with the plant. Plant, that material is different. Our indigenous cattle, that material is different. Here we are having one generation, means from one animal, at least 4 to 4.5 years. In indigenous, nearly 4 years, 3.5 years. In crossbred, nearly 3 years to get the progenies. That time there is no comparison with the plant. Here we have to conserve, we have to improve by the selective breeding, and non-descript population that should be improved by the use of good quality of the semen for upgrading of non-descript population. That is from my side. There is no problem. We can implement the selective breeding. There will be no problem. And I have already visited that place, Pune, as well as that Baramati. Dr. Mane, he was already with me. I have seen so many farmers, they are already maintaining the Sahiwal, wonderful Sahiwal. If you visit the Punjab, I think from my side, they were having the excellent phenotype, excellent Sahiwal animals. Nearly 700 to 800 animals available in that tract. Why they are maintaining? Because they are getting the benefits. So why not? We have to work on it. We have to maintain, we have to conserve, we have to improve our indigenous cattle. 50 years back, crossbreeding was started. That time, our target was to improve our non-descript population. But what then? We have already crossed our indigenous with the HF and Jersey, and now we have lost our indigenous population. So that still we have to go for the improvement of our indigenous cattle by selective breeding, and then we can go for the grading of non-descript cattle. All from my side. There is no problem. Farmers will get the benefit. Naturally, it will be sustainable. Our duty is to provide the benefits, provide the support to the farmers, stakeholders. They are already ready to accept our these facilities, our this concept, and all these things. They are ready. There is no problem. Thank you very much. And semen is available. If you want, we can provide it. Thank you all. Thank you. Thank you very much for giving me the opportunities. That Honorable Vice Chancellor and organizers. Thank you all. I was not available because I was at the headquarters. Some election is going on, so that I little bit joined late. Sorry for that. Thank you, sir. Thank you. Thank you.
Thank you, sir. Thank you.
I want to add some comments.
Yes, sir.
Yeah. Like we are talking about the non-descript animal or non-descript animal, it is not like this. This is my humble request. First, we identify the unique population among the non-descript animals. And this unique population may be characterized, evaluated, and we need to develop the breed descriptors. And if found suitable, then we need to register them as a distinct breed. It is the only way to reduce the proportion of the non-descript animals. Recently, we have submitted a project to ICR. It costed around 86 crore, in which our objective is in whole of the country. First, we need to identify unique population, then characterize them, and if found suitable, then register them as a distinct breeds. And whatever is left, we will say them a mixed population or mixed type of population. Then these mixed populations can be upgraded by the grading of with the well-established breed. And we are also submitted the proposal to study these mixed population, to study the genetic architecture, to understand the genetic architecture of the mixed population. Further, if we find something, then we will suggest that this population need to be upgraded by that particular breed. But that project we have recently submitted to. If this project is approved at this moment, the description of the non-descript livestock, it is priority area. I mean, our Honorable Prime Minister has mentioned in his speech, and in every meeting it is coming up. So once this project gets sanctioned, then NBGR will run this project in whole of the country with the help of the state departments, state agriculture universities. Then I think it would be a good work and good attempt. Thank you.
Okay, thank you. Lastly, I would like to request Dr. Nitin Martande for your views onto this topic.
I want to ask one question to Dr. Pundir.
Yes, sir.
Yes, please.
Now we are doing the breed livestock census. Have we supplied the breed descriptors to the Animal Husbandry Department for doing the breed-wise livestock census based on breed descriptor?
In 2013, we have supplied, and now whatever the breed registered, we are supplying these breed descriptors to the Animal Husbandry Department.
Because unless and until the breed descriptors are used,
we are supplying, and we are also requesting them if they need some training for trainers. We will also provide.
That is very essential, because the people who are doing the livestock census, they are not aware about the breed descriptor, the characteristics of the different breeds. Physically supplying the breed descriptor to the Animal Husbandry Department is different, but training the people who are doing the livestock census, they should understand that what are the characteristics of the different breeds. Then and then they will be able to do it properly.
We are requesting them for training also, and
I
we
think
are
that
also
training is
providing
very
breed
important aspect, because the people who are doing the livestock census, they are from the last cadre, right? So
yeah, this is the problem. I give you one example in this regard.
Yeah.
In Uttarakhand, they mentioned that rats in the cattle.
Yeah.
Population is around 5 lakhs. So we have taken a research project for rats in the cattle. Then we went to the state departments and discussed with the director. There are no animal in the field. From where these 5 lakhs animals are coming up? So finally, they come up that whatever the red color animal, they have mentioned it, the red sea. So these are the problems, and slowly we are improving that.
Because breed-wise census, even if we do it, this will be misleading, as we have pointed it out. So I think that this is a very important area on which we all over the country, especially NBGR, should emphasize on this.
No, we are doing it.
There could be a training program for all the field workers who are doing the census.
We are requesting him training for trainers.
Yeah.
Then they will train them.
Right.
Dr. Dheeraj Kankar, you can proceed.
One question is in my mind, Dr. Pundir sir.
Yes, sir.
Because in natural farming, all peoples are saying that you use the deshi cow dung, deshi cow's urine, and data as no, don't use the crossbreed cow. That's why. Is it data available? Because we have no data. Because what is the difference between deshi cow and crossbreed cows dung and urine? And why is it available? Please highlight this aspect in front of this. This is not subject, but it is a doubt in mind. That's why we are clarifying in this aspect.
Scientific data is missing on these grounds. Scientific data is not there. There are few persons. Those are making it. This is organic farming, and one cow can go for 30 acres of the land farming. Like this is general perception, but scientific data is not there.
Okay. Second thing is the deshi cow's ghee and crossbreed cow's ghee. Ghee is prepared from after removing the fat, and that's why there is. I think there should not be difference between this. Only procedure is different. For removing the creams and preparation procedure is different. But is it there difference between deshi cows and crossbreed cows' ghees?
Are you talking about A1, A1, A2, A2?
No, not A1, A2. Because we are
fat content is different. In deshi cow, fat content is higher. In crossbred, it is less. Fat content is less.
No. Fat is different. Fat percentage in this because after removing the ghee, at that time fat is 100% fat. There is 100% fat.
In ghee, there is no protein. Only fat. In ghee, there is only fat. So there is no difference.
What difference between deshi cows and crossbreed cows' ghees? When preparing butter,
fat is fat.
Yeah.
Fat is fat. Protein can be different, but only fat is fat. If you look the protein in indigenous cattle and this crossbred cattle, there may be different.
People are giving more money to the deshi cow's ghees.
In fat or ghee, there is no difference.
Yeah. Okay.
Fat is fat.
Yeah.
In case of protein, even A1, A2, also it is a controversial issue. Some are saying that A2 is favorable, but there is no proof that A1 is harmful.
Yeah.
This is again it is a controversial issue.
You are seeing the A1, A2.
Yes. It is again controversial issue.
Yeah, there is controversy.
Controversial issue.
We are observing in our dairy industries, we have so many samples we have collected of crossbreed cows. Only 10% cows showing the A1, A1. But as 80%, more than 80, 90, 85%.
No. All our indigenous cattle, buffalo, goat, all are A2, A2 type.
Yeah. Okay. But
even in crossbred also, I think 70% may be A2, A2 type.
Yeah, about 80%.
If you look the gene frequency, around 70% there will be A2.
Exotic also, sir. Many bulls we are A2. Exotic bulls also, Jersey HF also. It is not that they are exclusively A1.
It can be, yes. It can be.
Because
frequency is less, but it can be.
Yeah. Okay. Somnath Manik, continue.
Yes, sir.
Next panelist.
Dr. Dheeraj.
Yes, sir. Thank you. It's all our enlightened views and fruitful discussion. This is an electrifying 1 hour of discussion. Now, with immense respect, I request our Honorable Vice Chancellor, Dr. Prashant Kumarji Patil. By his kind gesture, we understand the words of Martin Luther King. That he said that same as applied with the thoughts of Dr. Prashant Kumarji Patil, Honorable Vice Chancellor. He always tell out of his gesture that if you have not discovered something you would die for, you are not fit to live. Sir, I request you to reveal the eminence by putting your views.
Good morning, good afternoon. Most respected guest of honor, of today's brainstorming session on conservation and improvement of indigenous cattle, Dr. Shyam Jawarji, National Mentor, Ministry of Fisheries, Animal Husbandry, Dairy, Government of India. Most respected Dr. Archana Verma, Principal Scientist and Head, Animal Genetics and Breeding of National Dairy Research Institute, Karnal. Respected Dr. R.K. Pundir, Principal Scientist, Animal Genetic Resources, National Bureau of Animal Genetic Resources, Karnal. Director of Research of our university, Dr. Sharad Gadagji. Dean Faculty of Agriculture, Dr. Pramod Rasalji. Associate Dean Sunil Masalkar. All the eminent panelists, participants, Dr. Umesh Singh ji, Dr. Aswal ji, and all the HODs from my university, and the very young team who is looking after this Indigenous Cattle Research cum Training Center, which includes Somnath Mane, Vishnu Naravde, Dheeraj Kankare, Pramod Sakre, Sunil Ranangale, and all my colleagues. It is indeed great pleasure for me for attending this brainstorming session, though I am not from the animal science background. But I thought initially that I will stay for some time. But after looking at the discussion and the seriousness in the presentation and overall conduct of this brainstorming session, I did not stop me that I should leave, so that I attended full session. And I am really thankful to all the guests of honors who have presented very nicely and complete scientific presentations with lot of information, and that I really appreciated. And Dr. Shyam Jawarji is having association with our university. He has remembered the years of 1983 when the Dr. D.K. Salunke was the Honorable Vice Chancellor, and he worked with our university. So that also get pride for us, and such a eminent personality with us today with us. So as you are aware that this Indigenous Cattle Research cum Training Center has been funded by the ministry or state government with a meager amount. And initial starting of this project, we can say, was a very, very slow pace. While taking over the Vice Chancellor, I thought that this is a very important project. And I have done some minor surgery in this complete project. Some people have been removed, some people have been added, and I am really very much satisfied with the kind of the work this small young team is doing in this establishing or implementation of this Indigenous Cattle Research cum Training Center is really appreciable. And you are aware that this with a lot of progress has made by the center. Almost we have now 8 Saiwal cows, almost 10 Gir cows, 6 Red Sindhi, 4 Tharparkar, 10 Rathi, almost herd of almost 40. This Desi Elite milk cows have been there now. And this is not only the training center, but this is actually scientific research also going on this indigenous cows. And we have IVF, almost 27 cows of MPKV Rauri Central Campus have been IVF done there. 10 cows at Agriculture College, Pune, and even for the farmers field, 25 farmers cow have been done with the IVF. So this is actually a really very important project, I can say, and this is on the right track. as you know, that integrated farming system and the climate change, these indigenous cows will be very immense useful for farming community, and because of that, they have very sturdy breeds, and you know, all of in Maharashtra there are so many clubs are going on now, Sahiwal Cup, Gir Cup, and there was demand also from the farmers that they want some proper education training in the so this project will be the actually very important for them, because this project is at the location at Pune, and they have gone behind they have doing a lot of activities. That IVF and ET work is going on in this project. Apart from that, they are doing the value-added products from cow dung, cow urine. Also, they have purchased silage making machine. Then various almost 10 to 15 milk products they have developed. Some are patented from this research center. And this will be actually a unique place for the farmers and the researchers for getting complete idea how this and Archana Verma is also told that sustainability is important. So from sustainability point of view, all the components of this cow project have been showcased there. And there also we are planning to have a Go Parikrama also, so that this is also one of the modern concept. Then cow cuddling concept will be also implemented, because worldwide it is being said that if you love the cow, if you are in contact with the cow, you touch the cow, all the stress related problems also will be healed. Such kind of innovative innovations also, innovative ideas also will be implemented in this project. So my plan is, because I am not the animal husbandry actually background, I am agriculture engineer by profession. So, but I am taking, I am monitoring this project actually day and night, so that this project will be at a very successful project, and it will be one of the best project in India. That it is my view. And I am now planning, if Dr. Zawarji will agree, we will have one monitoring team also from the ministry. I am planning to, if they agree, so that the third party evaluation guidance will be sought from time to time, so that this project will be on a right track. So I think Dr. Sham Zawarji, if you are agree that to be chair the committee, which will be monitored routinely, in which NDRI experts, or maybe Dr. Archana Verma, or maybe Pundir sir will be associated with them. Some other people also will be associated with them. They will, we will have a collaboration with you, and you can monitor our project, and whatever suggestions, corrections required to take this project scientifically will be very, I will be very happy that you accept my invitation for this monitoring team. And you are aware that you are aware that this is a very important project. I will not take much time, but very important point is here in this that there are, I think, almost that some of the breeds we have to add it, that can also be suggested, so that we can take this project ahead. And some funding from the Rashtriya Gokul Mission, if we get it, that it will be of immense useful for the running of this project. We are planning to make this project self-sustainable, first of all, and sub-funding from Rashtriya Gokul Mission for some particular activities, if you deem fit, which will be very much appreciated. With this word, I will not take much of time, but I appreciate the way the brainstorming session has conducted, and the seriousness in this, every speaker, every presenter, it is seldomly found in many cases, but this today's session is really a wonderful session with full of science, full of information, and I place on record all the participants who have expressed their views, maybe Dr. Umesh Singh, maybe as well Mr. Ulmekji and Dr. Gadak sir, and the way Shiv Kumar Patil, the way it has been conducted and the way it has been done, it is excellently organized session. With this word, I place on record once again all the presenters that for accepting our invitation and sparing their valuable time out of their busy schedule and presented this their views in this seminar. And once again, I thank all for attending this session. Thank you very much.
Thank you, sir, for showing the
Sir,
great.
I just want to add
Yes,
a
sir.
word. Honorable Vice Chancellor, my services will be always at your disposal. That's what I wanted to inform him. Thank you.
Thank you. Thank you.
Thank you, sir. Thank you. The magnanimity shown by our Honorable Vice Chancellor. Thank you, sir, for your energy and future way. And you're expecting us a lot of from us, and we'll sure the way you are supporting us, we will deliver like anything. Thank you. Now I like to call upon head of our department, Dr. D.K. Kamble, sir, for vote of thanks. Dr. D.K. Kamble, sir. He may having some
Just
sort.
not,
He may having some sort of a technical issue. I'm very grateful.
Sir, Kankare sir,
Yes, sir.
actually D.K. Kamble sir unmuted themselves.
Yes, sir.
Kamble sir, can you hear us?
Yes, sir.
Yeah. Actually, I have
Yes, sir.
promoted you to a panelist. Can you please accept the invitation, please, sir?
Yes, sir. Okay, sir.
अनम्यूट आहे नाही
आवाज काही
Actually, you are declining.
Hello?
Sir. Yeah.
Hello?
Yeah,
Yes,
we
sir.
can
Hello.
hear you.
You continue, sir.
येते ऐकायला
Oh,
Please go ahead.
Katkar sir? Yes,
Yes,
sir.
sir. Yes, sir. Yes, sir.
Yes, good afternoon to all. Respected Dr. P.G. Patil, Honorable Vice Chancellor, Mahatma Philip Dr. Rahuri, Dr. S.R. Dr. P.N. Rasal sir, Dean Faculty of Agriculture and Director of Instruction, Dr. R.K. Pundir sir, Principal Scientist, Animal Genetic Researches, ICR, NVGRA Colonel, Dr. Archana Verma, Principal Scientist and Head, Animal Genetics and Breeding, ICRA, NDRA Colonel, Dr. Shyam Johar, National Mentor, Minister of Fisheries, Animal Husbandry and Dairy, Government of India, Dr. B.R. Ulmekji sir, ex-Dean, Faculty of Agriculture and Director of Instruction, Dr. Sunil Masalkar, Associate Dean, College of Agriculture, Pune, and all participants. The team working on Indigenous Cattle Research Club Training Center, Division of Animal Husbandry and Dairy Science, College of Agriculture, Pune, organized one-day brainstorming session of conservation and improvement of indigenous cattle by the suggestion of our Honorable President. I am very much thankful to Dr. P.G. Patil sir, Honorable Vice Chancellor, for giving valuable times, even though his busy schedule. Now, I am very much thankful to Dr. Sharad Gadak sir for narrating the importance of today's topic. I am very much thankful to Dr. Rasal sir, Dr. Ulmekji sir, Dr. Thomre sir, Dr. Mandakule sir, Dr. Markandev sir for giving their valuable time for this session, for this brainstorming session. Also, I am very much thankful to guest honors, Dr. R.K. Pundir sir, Dr. Archana Verma, Dr. Shyam Johar, who is giving valuable information on the topic of indigenous cattle, genetic research and their conservation, breeding strategies for conservation of the indigenous cattle, and role of IVF in conservation of indigenous cattle, respectively. I also thankful to all the associate, also thankful to Associate Dean Dr. Mahaswag sir and all faculty members working in the projects conservation of cattle and research training center, Dr. Kankari sir, Dr. Mani sir, and their supporting staff. Thank you, sir. Thank you all.
Thank you, sir. With the permission of President and the Chairman of the session, Dr. Prashant Kumarji Patil, on behalf of him, I declare the session is over. Thank you very much.
Thank you, sir.
Thank you.
Thank you, sir. Thank you. क्या आपको रूप चलते रहते हैं
बहुकूर्वित तदव्रतम् जय 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 हरित क्रांति जय
देखिए अमृत काही बढ़िया आहे. अरे
दीपक आवाज येत नाही. दीपक आवाज येत नाही.
Hello.